Okay, great. We'd like to welcome everybody to come in and have a seat as we start the Human Cell Atlas General Meeting. Welcome, everybody. Welcome to Toronto. We are here in the Mars Discovery District. This is a tech and biotech campus next to the University of Toronto. And all of the details about the meeting are on the event site that everybody should have. And we'd like to invite people to tag on Twitter, HCA 2023 GM, if you want to tweet so that everybody can see and follow along at home. Just a quick notice that there will be filming in all of the venues in person and virtually. And this, everyone should agree for that. If you have any questions, you can contact meetings at humancellatlas.org. I forgot to introduce myself. I'm Gary Bader, computational biologist in Toronto, and happy to be the host today. To start, I wanted to acknowledge that this land that the University of Toronto and the Mars Discovery District operates I want to acknowledge the land. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. And today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on the land. This land acknowledgement is a formal statement recognizing the unique and enduring relationship that exists between Indigenous people and their traditional territories. And for people that are not familiar with this idea of land acknowledgement, we've put some resources together on the website. So if you go to the resources section and click on land acknowledgement slides, you can get a whole historical background about this idea and learn more about it. We'd like to thank a number of sponsors for very generous donations to make this meeting possible. In particular, the Helmsley Charitable Trust for their foundational level support for this meeting, as well as gold level support from the University Health Network. It's a big research institute here. Half of this Mars Discovery District is filled with UHN labs. And as well as Medicine by Design and Princess Margaret Cancer Center for their silver level support. Again, most of them are in this building, in this Discovery District. And also CFAR, the Canadian Institute for Advanced Research, the Company of Biologists, the Sick Kids Research Institute, the University of Toronto McLaughlin Centre, and the Ontario Institute for Cancer Research, which is another research institute in the centre, for bronze level support. And the HCA in general is grateful to operational support to make the whole thing run from the Chan Zuckerberg Initiative, the Klarman Family Foundation, and the Wellcome Trust. So thank you very much. So I'm going to start by just providing a little bit of information about the venue and the rules around here, and then we'll talk a little bit more about the meeting before we start. So we have health and safety set up here, which is great. So we have safety officers John Randell. Do you want to put your hand up? And Shamini Ayaduri, who is around too. They're putting their hand up. So they can be reached at safety@humancellatlas.org with any questions you have about health and safety. Ensure you're wearing your name badge so that you can get access to meals and the meeting venues smoothly, and as people will be checking. Everyone should feel safe here, so please respect all the different preferences regarding physical proximity due to COVID concerns. So we have a system that you probably heard about where the lanyard color represents people's personal preferences with physical proximity, with red meaning please don't come close. Yellow is elbows only, or fists maybe. Green is hugs and high fives are fine, and handshakes. And you can switch that around if you'd like at the registration desk during the meeting. Of course, if you have any symptoms of COVID, please stay home from the meeting. There is a virtual platform, and it should be easy to connect virtually if that occurs. Any other questions, visit the registration desk or email meetings@humancellatlas.org. There is a meetings code of conduct, which is posted on the HCA website that is also in effect, that covers the policy of the HCA around inclusivity, accessibility, respect, and a harassment policy, and the consequences of not following that policy. Again, if you have any questions, just reach out, and you can look online for the details. And we take that very seriously, so we'd like to make everyone aware of it. So situating ourselves here in the auditorium, there are a couple of places that the meeting is going to be held at. So in particular, for some of the breakout rooms, as well as some lunch seating in a cafe, we have to go upstairs, up the escalators, and to the right. And there's a place called the Collaboration Center that you have to go through some doors, and then there'll be a bunch of rooms in there. We have all of those rooms for breakout sessions and lunch seating. The posters will also be upstairs in the atrium, and so you'll access those as well from the escalators. Other things, Wi-Fi is free, Mars guest, there's no password needed. Food and beverage, we're very lucky. You can just eat anywhere. You don't have to worry about food in the meeting rooms. You can bring it in here. And just make sure that you don't leave a mess. This is a public space with a lot of hospitals and businesses in this center, so it's not safe to just leave a laptop lying around, although it'll probably be fine. But because we're in a public building, we're just advising people to make sure to keep track of their belongings. There is a luggage room that you can access if needed to lock important belongings on a first-come, first-served basis. So with that preamble out of the way, I'd like to thank the program committee as well for putting together the scientific program. Myself, Marioni, who I haven't seen this morning but is there, hi. Dana Payer, who unfortunately at the last minute her flight got canceled due to storms on the East Coast, but we'll have her virtually. Shyam also had a similar problem. Aviv is here, and Sarah will also be virtual, unfortunately, due to COVID. But we wanted to thank everybody on the program committee for putting together a fantastic scientific program. Anybody can contribute to the Cell Atlas, and we are really encouraging everyone to think about how they can contribute. For everyone here who's a first time, and we'd like to encourage you to learn about the HCA and its history and how to contribute. One way you can do that is by joining a biological network. As a member of a biological network, you can help select data sets for integration, implement the roadmaps towards building the atlas, participate in cell type annotation jamborees, analyze and integrate data, and join meetings and collaborate and meet a lot of researchers who are working in an area that you're interested in. There are 18 biological networks focused mostly on major organs of the human body, and biological network coordinators are marked on their name badges and are listed on the HCA website, so you can email them. If you want to meet at the meeting, you can, a good place is the registration desk to set up meetings. Also, if someone is part of a biological network, they should have a little ribbon on their badge with the name of the biological network, and if you want to get those at the front desk, at the registration desk, you're welcome to pick up more. So that will help us find each other at the meeting. More information about the HCA is on the humancellatlas.org website. So together, we want to build this atlas, and the overall meeting goals today are, of this meeting, are to plan the continued work on building the atlas version 1.0. There's a lot of interesting discussions to be held about that topic. We also want to really focus a lot on imagining future atlas versions. What interesting new dimensions do we need to work on to eventually build a map of all the cells in the human body? We also have an important topic weaved into the program here, which is connecting with the machine learning community. The Vector Institute is a machine learning research institute in Toronto, and Mila is a similar one in Montreal, and we have representatives from both here. Vector is also in this Mars Discovery District, and we've integrated that into the meeting program, so because we know there's going to be massive amounts of data produced, already is, and we need to really work to figure out how to analyze that. New members are noted on their name badges if you picked up a first meeting badge. And so if anybody who already knows about the HCA sees someone who has a first meeting badge, please reach out and talk to them, find out how their interest in building the atlas. Virtual attendees, we have, I think, 700 virtual attendees, I think. It's amazing. We really hope virtual attendees out there can engage with the meeting. We have ability to receive questions and chats, so please make use of that proactively. We are really looking forward to hearing from you and communicating with you virtually. Some virtual attendees are presenting. For those people, to engage with them, please find their email via the meeting website profiles and offer to contribute where you see a need. If you learn about what's happening in the human cell atlas, it's a huge project, and you're interested, even if you're already working on it, I think that this is one of the goals of the meeting is to coordinate, find out what everyone's doing, what's needed, what are the gaps, how can we fill those gaps and move forward. So we hope that there will be lots of that at the meeting. So the last point I wanted to make is just a review of the schedule today. We'll have plenary sessions before lunch. And during lunch, there's an equity working group meeting in CR2 up the escalators to the right. Breakout sessions, 2 of them will be upstairs in the collaboration room, and 1 of them will be in this main hall or the auditorium. It's the same thing. Lightning sessions, lightning talks in the afternoon, and then a group picture, and then the poster session upstairs with a reception. So that'll be a really fun way to connect. So. With that, I'd like to welcome Aviv to the stage and Sarah virtually, if she's connecting. And so Aviv, welcome. Aviv Regev, who is the co-chair of the Human Cell Atlas, is going to present an overview of the Human Cell Atlas.
Okay, hang on, this got stuck. Hopefully it works now. Yes. Okay, so first of all, a big thank you to Gary and everyone locally here in Toronto. We actually had to pivot to Toronto. It wasn't our plan for this year initially. And Gary had to put this meeting in literally half the time that we usually do that. And it's clearly been an absolutely amazing experience working on that. So a big thank you to Gary, and you should all. As Gary told you, unfortunately, Sarah has COVID. She's probably the last person on earth who hasn't had COVID before. And it's not fun having COVID, but you also can't travel. He also pointed out that there were thunderstorms yesterday on the East Coast. And anyone who's ever lived in Boston or New York City knows if they say thunderstorms, it means the airport is going to be a zoo and all the flights are going to get canceled. So Dana and Shyam are both stranded at the moment. They have not managed to make their way through here, and they'll join virtually for that reason. And so welcome everyone who's here, everyone who's virtually to the HCA General Meeting. It's really fun to see everybody. Usually I get to split this talk with Sarah. I'm going to present alone because we wanted to make sure that this works smoothly, but she'll be here for the Q&A and throughout the meeting in many ways. And we also wish her, healthy, much health and a very fast recovery. Okay. So in this opening talk, I'm going to give you a very brief update on what's up at HCA, but I'm going to especially focus on how, during the meeting, we're going to be discussing these big questions that are looming for us right now as we're grappling with how we pursue the mission of the HCA, which is to create comprehensive reference maps of the types and properties of all human cells as a way, a basis for understanding, diagnosing, monitoring, and treating in health and disease. The HCA community has grown and it continues growing. This was a pretty dramatic process over the last several years since we launched the Atlas collection. And what I think we're most proud of right now is that now it's a truly global footprint, and that includes the global South. So we now have leadership and a really vibrant and growing membership across the globe, including in South America, in Africa, and in the Middle East, 3 areas where we didn't have as many members when we just launched the HCA. And together with the members that we have in North America and in Europe and in Australia and in Asia, this means that when we build an atlas, it's diverse, both in terms of the scientists and in terms of the data, and that people are really working in the communities that we aim to serve. Overall, we just passed the 3,000 member mark. You know, it's a real number because it's not 3,000, it's 3,009, and from 95 countries and over 1,500 institutes. Now, there's many ways in which we're organized, but one of the main ones is that people are organized in these 18 biological networks that Gary described to you as well. And that's really where the data gets collected. Each of these networks is generally focused on an organ or a system, but we also have a couple of networks that are devoted to overarching goals. For example, we have a network for genetic diversity to make sure that we're creating an atlas that's diverse genetically and ancestrally. We also have a network that's focused on human development, which is something that happens across the entire body and you can't really parse just by organs and systems. Across all of these networks, there's constantly data growth in terms of the number of individual samples, the cells, the numbers of samples from each individual. And I'm just going to pick on a few highlights because it's impossible to go through all of them. So for example, we've seen a ton of progress around the nervous system and in particular the brain. And this is really due to the fact that there is a major initiative funded by the NIH called the Brain Initiative. And Ed Lein, who's somewhere here, I saw him this morning, is going to talk today, this morning, a little bit more about what's happening there, and in particular how things are even picking up more speed right now as the BICAN initiative has been started in order to build the ultimate human brain cell atlas. Another major effort is in the lung. This is also our first released integrated atlas. I'm going to talk a little bit more about this soon. But there's a few others that really stick out in big numbers. One of the most prominent ones is the immune one. Partly, this is because peripheral blood mononuclear cells, or PBMCs, as well as whole blood, are, kind of easy to get from people. So you get a large number of those samples. But the second part of this is because immune cells are everywhere. So pretty much in every network, when we collect data, immune cells come along for the ride, and honestly, the tissue wouldn't have survived without them. So that's, again, a very large effort. This actually segs into also the effort to collect a genetically and ancestrally and geographically diverse atlas, which is also rapidly growing. Many of the first efforts on genetic diversity are focused on the immune system, and Chloe and Shyam, in some tag team effort, I'm not sure how we'll do it technically today, given that Shyam is one of our stranded participants, are going to talk to you a little bit about that this morning. It's also actually really exciting to see how rapidly other efforts have been progressing. So, for example, the oral and craniofacial network and the adipose network, they weren't as prominent or as active a few years ago when some of these other efforts have been going, but both of them have been progressing really fast. Or if you look at the reproduction network, some of the organs and systems that the team there is focused on are things that are actually very understudied in biology and that lead to a lot of unmet need. For example, endometriosis in females is related, of course, to our reproductive system. And so across the board, really exciting. And this is stopped moving. Let's see why. Hopefully now it moves, yeah. And not only are the networks hard at work at collecting data, they're also trying to build these frameworks, which we call roadmaps, to guide the work ahead. So 4 of these roadmaps have been completed and published for lung, gut, oral and craniofacial, and for development. More are forthcoming, and all of these are going to be reflected in the overall white paper that we're in the process of revising. It's also a great opportunity for me to just thank the coordinators of each of the networks. They work tirelessly and they work enthusiastically, and it's a lot of work to keep this going, and so we're very grateful for them. Okay. So now, with all of these data growing rapidly, we of course have pivoted in the past year into what we call Atlas integration, which is also the first theme of our general meeting. Gary highlighted it for you as well. And in this morning's session in particular, we're going to be focused on what integration actually looks like. What does it look like now, and what do we want it to look like in the future, and what are the challenges between the now and that future. And so, as you may recall, as you may recall, in the previous general meeting, we kicked off a multi-group team to start on this effort. The first step was to pilot what we call the minimal viable product, or an MVP, of integration, and we chose 6 systems in what we called wave 1, the immune system, kidney, eye, gut, the nervous system, and the lung. So when we chose these, we had several different kinds of criteria. So for example, we wanted to have available and mature enough data to do an integration. We needed a committed set of bionetwork coordinators that were willing to partner in this effort. And also, we needed these to reflect somehow different kinds of organs or systems, and so different kinds of integration challenges. So we're not just repeating the same thing again and again and again. And Malte Luecken is going to give us an update on this initial effort. Now, some of the integrations actually already have kind of their MVP pretty much there. I'm highlighting some images from the retina and from the lung. Others are ongoing, but everything is progressing really nicely. I promised you a little bit more about the lung because this was actually a first great example for us of what integration could look like, and also inspired us in feeling that we can really set to work on this across many different systems. And so the first integrated human lung cell atlas has just been published about a week ago, I think. Yes, Fabian, 2 weeks ago. About 2 weeks ago. And this atlas has a harmonized cell annotation ontology. It has harmonized metadata. It has diverse anatomic locations in an initial common coordinate framework for the lung. So it's a good substrate. And then the subset of the data served to build the core integration, and then it was extended with additional data sets using transfer learning, so that there's a total of, 2.5 million cells roughly, 50 different data sets, almost 500 individuals. These are pretty robust numbers. And then the team showed also how you would use an integrated atlas like this. You can query it in all sorts of ways. You can generate detailed cell annotations and markers. You can look at the effect of different parameters like age or BMI or gender on cellular profiles. You can relate GWAS genes to cells. You can discover new cell types. You can look for transitional states. Not everything is a cell type. And so this and many, many more, go read the paper, but also a nice template for us as we think about additional challenges in other networks. And so kudos to many, many people, Lisa, Malte, Fabian, Pascal, Jay, Sasha, Martijn as coordinators, but also the whole network and the many, many contributing labs. Now, there's a lot of work that remains to be done. First, to finish the first wave MVPs, and then to move them from minimal to maximal. But at the same time, we're kicking off 6 additional integrations: the heart, the liver, the skin, the pancreas, oral and craniofacial networks. These are all kind of similar. It's an organ or a system, but also the genetic diversity network, which does pose quite distinct challenges and is a very exciting move for HCA. Now, underlying any integration are 2 things. There are scientists and there are human expertise, and there's computational tools and algorithms. And there's a large space of tools right now that are emerging in our community to do that. These are just a few illustrative examples that I think people are using quite heavily, and those include tools to generate references, to query them, to annotate cells, and to expand them as additional data is added. And regardless of the computational tool that you use, we need to put the knowledge that we have in the end into the right type of software platform. And so as the integrated atlases are getting built, our cell annotation platform, or CAP, led by Chloe Villani, who will speak later this morning, helps investigators create a reference for the cell identities based on molecular signatures. And instead of just making this a static place where somebody puts in the annotations, this is a living, breathing kind of platform, so that the platform is centralized, but it's really driven by the community that creates and explores and stores and serves these annotations at multiple levels of granularity, so that different people can provide different annotations. You can collect all of the information, and over time you can also train algorithms to do some of this work. And so it's really exciting to describe progress, but it's also important to remember that the key most word in a minimal viable product is that it's really minimal. And that means that there's a lot of difficult things ahead of us, and we should absolutely not be satisfied with what we have in hand right now. So I wanted to highlight some of the open questions that we feel we really need to tackle in this coming year or couple of years as we build this first Atlas version. And one that is top of mind right now, and I already alluded to, is the concept of defining integration between the clean and simple, strict definition of cell types. So saying all of these cells belong to a category and they have a name is an important thing. We use it, it's important for us. Things that have a name and are categorized are things that we can pursue. But in no way does that begin even to describe all the ways in which you can think about a cell. And in particular, there's many things that just don't follow these strict boundaries. And so, for example, thinking through the lens of gene programs can complement the way of cell types, but it is not yet fully reflected in these integration efforts, although there's a lot of research that goes around that. A second major challenge is balancing between the desire to harmonize things and make them look the same, and the desire to capture the inherent variation that is always part of biology. And that's a variation between conditions, locations, individuals, and so on. And what's important to understand is there's no, and there's never gonna be, one correct answer. It depends on the question that we ask. So if we want to build a simple reference, for example, define all the T cells across individuals, all the T cells in the body across individuals, you want to remove some of the biological variation, for example, between different people. But if you want to study inter-individual variation, for example, you're interested in human genetics, then you want to parse out what's common features that let us first relate all of these cells as T cells and preserve the variation between the individuals. And this problem becomes even more prominent when we consider going across tissues and integrating the ultimate human cell atlas, which is actually a whole body integration. Here we know up front that cells, for example, T cells, are the same and different at the same time. So immune, stroma, even parenchymal cells like epithelial cells, they have common features across tissues, and they also have tissue-specific characteristics. So if you try to categorize everything just to one strict category with a one-to-one matching of a cell to a type, that's no longer going to cut it. And we need a more flexible framework. Oops, sorry. I lost something. There we go. Okay, so all these questions become even, all these questions across all of these topics, kind of that's what looms under or above the word annotation and how we actually do it. How do we balance between specific experts, the community as a whole, what algorithms can do for us automatically, how do we bring all of these things together, and how do we annotate besides discrete cell types? And so in session 2 this morning, and in the first breakout, we're going to be covering many of these questions through presentations. We have a panel, I believe, and we have all sorts of open discussions in the breakouts. Now, after these come the many, many things that this first wave integration effort hasn't yet taken on, either deeply or even at all. So, for example, the challenge of integrating data that represent dynamic and developmental processes is not one we've done yet. We need to, but we haven't exactly done yet as a full integration effort. Buzz is looking at me like this, but we need to do it, it's hard. And then moving from molecular cellular profiles to other levels, especially spatial ones, is another one that we haven't taken on yet. Not as a community, we do this in HCA, but as an integration effort. And so this brings me to the second major theme, which is Atlas expansion. So HCA was never about RNA profiles of cells. So I chose to myself the little tag that says first HCA meeting, not because this is my first HCA meeting, it's not, but because I was at the first HCA meeting. And even then, in 2016, how shall I put it? It wasn't only about single-cell RNA-seq then either. You can go, well, we didn't stream that meeting. We only started streaming 2 meetings down from there. But we have all the materials. People were already talking about all the things we are talking about now, even though we didn't have great tools yet, lab or computational for some of them. So it was always about all the features and properties of human cells. And these additional features are now taking center stage. So yes, single-cell RNA-seq gave us a fantastic first start. We're very grateful to it. But the cellular branch of the atlas, as we called it in the white paper, it includes many other features that are measured at single-cell resolution. I don't know, you have your chromatin organization, your protein levels, your protein modifications, your metabolic profiles, your microscopic images with imaging, dynamic events. We can measure today's transcription rates, translation rates, and then thanks to multiomics, you can also measure all of them simultaneously in one cell. And then on top of that, to understand cells truly, we have to know where they're located, both with respect to some abstract common coordinate system, or framework, CCF, and with respect to each other in the tissue. And so this is what we call the spatial branch of the atlas. And now there's been many technical and algorithmic advances, and it's progressing a lot more rapidly. And then finally, on top of all of these, for this to be a human cell atlas, that atlas must reflect human diversity, and that's in terms of ancestry, gender, age, and geography, and many other things as well. And so this first integration effort looks just at one view of cells based on RNA profiles. At the same time, the community is pushing along to collect on all these other dimensions. And as these additional views are growing, we have the opportunity not just to integrate, but to unify. That means to build a unified representation and a unified understanding of what it actually means to be a cell. And that's very different than just saying a cell has these and a cell has that. You want to connect these different views. You want to be able to computationally learn how to predict one view from another. And you ultimately want to understand how one level of organization yields another level of organization in a cell. And so we're going to spend a lot of time in the coming days on this atlas expansion. Session 6, for example, is going to focus on spatial methods. Session 7 is going to look at how adding these new dimensions helps answering new types of questions. And in session 8, we're going to look at how machine learning in particular, to Gary's point about the great opportunities with Mila and Vector, can help us integrate not just across data sets in one modality, like in this first wave, but also across these levels to unify this atlas. Okay. So now as this atlas grows, we should care not just about how we assemble it, but how people use it. And the core question is how we query the atlas to help us answer questions, and that's the third theme of this meeting. And so, as in many other data-rich fields, the scale of our HCA data opened the door for us now to learn and query foundational models of cells across the human body. And we're starting, hopefully this works, and we're starting to see these kinds of models emerging now pretty quickly. And they're relying on different types of machine learning paradigms. You have work with transformer-based large language models. You have work with metric learnings. You have all sorts of things. What matters is that we need to think about how we can harness the tools for what we want to do. And so we're going to spend some time on how we can use these frontiers, these computational frontiers, in order to help us assemble and query a cross-body human cell atlas. And we're going to do this in session 8, focused on foundational models, in session 11, on where the frontiers of machine learning meet the human cell atlas, and in breakout session 12 on building the whole body atlas. And being here in Canada, and this partnership with Vector and Mila really helps us bring, we hope, the best minds to think about this problem. Okay. And then finally, this time at this meeting, we're actually pushing beyond the scope and remit of the atlas in the strict sense, and we're going to think on how the atlas, as well as the techniques that our community develops, can help move from descriptive cell annotations to causal understanding of cell function. And so think about it like this. Right now, strictly speaking, from the kind of data that you could collect from, say, single cell RNA-seq of some tissue, and the kinds of tools that we have, you can readily ID cells, you can categorize them, you can label them with all sorts of names or give them a new name if no one has seen them before. You can say where they are at different levels of resolution, and you can describe in which conditions you found them, healthy or disease. All of these are extremely useful things. But unless you already happen to know what a CD8 T cell does, how it recognizes another cell and then how it kills it, our data, that kind of profile, wouldn't actually tell you that that is the cell's function. We happen to know it from before. And it's useful to say this is that, and it has all sorts of clues, but it doesn't give us function. And so at the close of this meeting, we're going to take the cell function question on. And as I said, this is not what the atlas is expected to deliver. We didn't promise this. This is not its goal. But it is something that both the atlas data itself and all of the methods that we're developing enables. And I also think it's something many, many of us care about very deeply. And I'd venture to say that it's probably one of the reasons most of the people in this room actually work on this, is that they want to understand what things actually do and what their function is. And so in session 14 in plenary, we're going to hear several perspectives on how we should think about cell function. And then for those who are interested further, we're going to dig deeper into this in part of the breakout sessions in session 15. Okay. Before I close, I want to spend a couple of minutes on the substantial efforts of all our working groups and committees that keep HCA going. It's a lot of work. We have 4 working groups: analysis, technology, ethics, and equity. Some of them will meet during the general meeting, and in particular, the ethics working group is welcoming all who are interested in this to attend their committee meeting and join them at the lunch break today. I also will highlight the work of our equity working group. I showed you already in the beginning that HCA has come a long way in making sure that we're a true global initiative, and this is thanks for the effort of this working group. That's how we can be diverse in our data and diverse in our scientists, and our scientists can do this work within the community where they live and serve. And this involves a lot of dedicated groundwork, and one of the nice things that happens is these workshops, which we've had this year in Latin America, in Asia, and in Africa, and soon we're going to have one in the Middle East, right? Yes. And so as a great example of those, one great example of those is this recent HCA Africa workshop that was put together in March. And in this meeting, this was a 4-day workshop. It brought together 60 experimental and computational biologists from 11 African countries. I don't know them by heart, but I wrote them down for myself. Ghana, Nigeria, Ethiopia, Zambia, Malawi, Mali, Cameroon, Kenya, Gambia, Senegal, and South Africa. And the participants were evenly split between computational and experimental. They had parallel tracks for computational and experimental activities, a lot of hands-on training, and so when participants left the workshop, they could bring back this work to their own labs and research. Now, all this work in HCA leads to a lot of papers, 128 to be exact, that are either published or in preprint form and already HCA branded by their authors. And that's done with the help of a diligent publication committee. And there's going to be more. And so next up, we have the 3 publication bundles with about 85 papers in them. And they've just started making their way through Nature Publishing Group. Keep your lookout for emails from Ellen. This bundle has 3 tracks, one for analytical methods, one for developmental biology, and one for organ or system-specific atlas integration. If you have any questions about the bundle, but also if you have a paper that's actually ready and you didn't sign up for the bundle and now you changed your mind and you would like to contribute, it's not too late. And you can email Ellen at the address here, or you can find her, I assume she's somewhere here. She is, and she has a very HCA-colored shirt to make it easy to find her. Okay, and then as always, HCA is just an open initiative. We don't have any money of our own that we hand out to people. I'm sure you all know that already. It's only possible because we have a nice partnership with many, many funders who are themselves part of HCA. And they support HCA researchers through many different initiatives and grants and mechanisms. And this is even more augmented by the collaborations that our scientists in many ways have in the context of disease atlases, because you can only understand health in the context of disease and vice versa. And so we're very grateful to all the funder partners. I'm very grateful to all of you for joining. It's so nice to be here. I wish everyone a wonderful general meeting and a fun 3 days. Thank you very much. And now I'm waiting for Gary to tell me what to do.
Thank you very much, Aviv. You were perfectly on time. It's amazing. I didn't have to do anything as a chair to work on that, so thank you very much. Do we do questions? Or we can do,
well, you're the chair.
Yeah.
I believe we can do questions, but we always run out of time.
A couple of questions before we move on to the next session, if there are any.
Yeah, you need to continue holding your hand up because they have to get you the mic because the virtual participants wouldn't hear you otherwise.
Okay, you want to go first? Okay.
Yeah.
Oh, there's online questions, yeah.
Yeah, we've got an online question from Dana. Gary, do you think the liver is the best atlas?
Dana is clearly in good
Where's my
form.
liver badge? Yes, and we will be giving out liver stickers to people who want.
I want the liver sticker.
And we will be hearing from a really great liver story later as well. One more question here.
Oh yeah, sure. So this is my first HCA meeting. I don't know if I'm out of my spot, but I just want to ask, I asked this in a virtual meeting before, but when you mention all human cells or all human cell types, how many would be those, or at least in an order of magnitude? In the thousands, in the millions, in the billions? Are 37 trillion cells on average in a human body? How many cell types is the atlas about?
So I'll take a stab, and I also see that we have Sarah online, and it's one of these foundational questions that's fun to muse about a little bit. So first of all, there's a big difference we now realize between the organs and systems. Probably the brain sits in a category separate from all the others in terms of its very fine resolution cell typing, discrete categories in very large numbers. In many other organs, you would have definitely over 100 or more in something like the gut, but then it gets complicated because of what I said, because of how they're similar and different at the same time. So you look at the small intestine and the large intestine, just choose a tissue I happen to like. And you can say, well, I have tuft cells in both. Or I have an epithelial stem cell in both, but it's kind of colored a little differently. Even within the small intestine, the three major parts of the small intestine, you will see a little bit of a difference in its epithelial cells. So do you count that as different things or as the same thing? Depending on how you do the counting, the numbers will start shifting quite rapidly. And this is why thinking just about the types is too narrow of a definition, and why we wrote, even in the beginning, the types and properties, because types is actually only one way in which you can look at cells. Having said that, I haven't been around during the Human Genome Project, okay? But one of the things you always hear from the old-timers is how bad it turned out for the people who made guesses on how many genes are in the human genome. So I promised myself before this even started that I'm personally never going to venture a guess. We'll just let I think the interesting question is, how much can we predict now, given the data that we have collected, how many more there are to go? And I'll turn to Sarah. And hopefully it works AV-wise.
Sorry, what was the question about cells?
A number of cell types, how many do you think there are?
Not a trick question. Well, we are, when we focus on individual tissues, like, the different tissues of the heart, the different tissues of the intestine, so Aviv has mentioned large, small intestine, different regions of small intestine, and so on. For each of those tissues within an organ, we're in the region of 100. For blood, we're sort of upwards of that. Of course, if you take together all of those tissues, in the heart, we're talking about 30 tissues, in the brain, 100, and so on. And we take the union, we're going to be at a much higher number. And that's really all I'm going to say on that topic for now, I think. I'm sure we'll hear more. We'll get more insights throughout the meeting during the days ahead.
Okay, thank you very much, everybody. I'd like to move on. Do we have one more question?
There.
Okay.
Yeah.
He was the first to raise his hand.
Okay, sorry, I didn't see that.
Sorry.
Go ahead.
I wanted to ask.
Actually, could you introduce yourself? I think that would be great for participants to say.
My name is Nada. I'm from Sarah's lab. Sarah and John, and so part of the HCA community, but it's my first meeting, so I'm happy to be here. I wanted to ask how, because there is one aspect that I think maybe is not addressed enough, which is kind of like the native state of the cells in the tissue, which we obviously, from obvious reasons, we extract the samples from deceased individuals or after immense processing. So how much do you think can we bridge that gap and how much of the function are we losing actually by the methods that we're using to probe the cells?
I think it's an awesome question, and it is addressable in multiple ways. There isn't a single answer. I think looking from multiple directions, it's like it allows you to triangulate where reality is, because all of them have some biases and skews. The first thing I'll say is that it really depends on the lab technique that we use. So in certain lab techniques, we introduce more difference from the native state in the tissue when it was obtained than with others. Dissociating cells is likely worse than extracting nuclei. Extracting nuclei is worse than doing it in situ in the tissue. As the methods have advanced, so are we getting closer to what we think is happening there. But the mere act of, excising a piece of tissue from an individual is going to make a difference. So that's one layer, I think, of difference. In some cases, we have the opportunity to obtain tissue that is probably completely healthy. So, for example, since we mentioned the gut, people go through routine colonoscopies after a certain age. Most of them are perfectly healthy. There's no reason for the colonoscopy except for a screening test. Those kinds of samples tend to be very reflective of healthy, quote-unquote, tissue. However, those usually tend to be limited. They won't be as deep, you won't see as much. There's a price you pay for that. Having both looks, having a look from postmortem tissue, from resections in surgery, from what we call adjacent healthy tissue in cancer resections, from biopsies, the more it looks consistent, the more you believe this is the native state. So that's a second whole pot of things. And the third one, which I alluded to a little bit when I talked about the different levels of organization, the more you relate different levels of organization, the more, again, you're closer to the unified actual latent space of what things really are versus the views of them that you see through the lens of measurements and so on. And in particular, if we get there, and I would say there's optimism that one would get there, you would get to the point where you might be able to use live methods and predict from them and generate from them what the molecular profiles are. And if you would be able to do that, that's, further in the future, but, one can dream. As you project like that and you go live, then you would be in a very different place than the one that we are at today, and also you would have better benefits for patients. That's why this multi-scale work is so fascinating and interesting. And in the meantime, we also get a lot of utility from the limited methods that we have today. I think that's a good place to stop.
Great. Thank you very much once again for a great state of the HCA address. Excellent. So our next session we hoped would be a display of some highlights from scientific advances that are being made based on single-cell genomics technology and the use of the HCA, all the data that's being generated as part of the ALICE projects. So I'd like to welcome our first speaker, Philip de Jager, who will be joining us online. And let's see if we can get that going. So, welcome, Philip.
Hello,
go ahead.
Yep. Hello. So again, I'd like to thank the organizers for inviting me to share some of our work with you today. And I'm really sorry that, like Sarah, is exposed to COVID and can't join you in person, for this outstanding conference. So I'll be talking to you about, some of how we've applied the lessons learned from the experimental and computational developments that the HCA has spearheaded, and to apply it to, in our case, Alzheimer's disease, as a case in point for the study of the central nervous system. So here are some of my disclosures. None of them are pertinent for this particular talk. One of the big challenges of studying the CNS, and Aviva just alluded to this in her introduction, is accessing the tissue, particularly in the healthy state. That's, of course, not feasible. There are neurosurgical procedures, but again, that's not healthy tissue either. But in the case of diseases like Alzheimer's disease, we have a lot of data, but there's an important divide between the antemortem data that we have access to during the life of the person and during the evolution of the disease from a pre-symptomatic to the symptomatic phase, which is shown here to the left, and we can do various things with blood, with spinal fluid, and with imaging. And then ultimately, of course, we can collect brains and then finally access the tissue and generate data with histology, and these days, of course, single-cell data. Now, one of the challenges and the focus of this particular talk will be how do we use this autopsy-derived data, cross-sectional data, to try to relate it to what was going on during the person's life? Can we infer trajectories that can then help us to understand the molecular mechanisms that are occurring during the life of the person so that ultimately, of course, we can intervene? This is possible because of colleagues who have developed a superb resource at Russian University, and I've been working with them for over the last 15 years. This is David Bennett, shown here at the bottom, and Julie Schneider. David designed and leads two superb cohorts of older individuals, the Religious Order Study, or ROS, and the Memory and Aging Project, or MAP. These are two studies of cognitive aging. Individuals are enrolled when they are non-demented, and importantly, they have to agree to donate their brain and spinal cord to the study when they pass away. And Dr. Schneider is the neuropathologist who's in charge of the brains. They've done a superb job over the last 30 years to collect over 2,000 brains with detailed antemortem data, including cognitive data, imaging, all sorts of functional data, and then postmortem, a very structured neuropathological assessment, and postmortem imaging as well. Again, I don't have time to get into all the details, but please reach out if you're interested in these cohorts. So working with two superb early career investigators, Naomi Habib and Vilas Menon, we set about to generate large-scale single-nucleus RNA-seq data from the aging neocortex, in particular the dorsolateral prefrontal cortex, a hub for cognitive and mood circuits in the brain. With a couple of technological notes that we did, so we actually used single-nucleus sequencing. We pooled nuclei from 4 individuals, and actually we didn't do any hashing. We just simply used the genotypes encoded in each cell's RNA-seq data to deconvolute the nuclei and assign them back to each individual subject. So this worked very well. And at the end of the day, we had 424 participants with over 1.6 million transcriptomes. What you can see here on the right side is the sort of high-level clusters of the different cell types of the neocortex, which are listed below. And of course, we then also went about to look at the different cell subtypes, of which there were 92 at the end of the day. Now, I just want to take a little detour into some of what we've done with these data. One of them was to map the effect of genetic variation on gene expression in each of the cell types, which you can see at the upper left corner here. The different cell types are noted here in green. We have, for example, the excitatory neurons. And what we're plotting here is the cell type proportion on the x-axis and the number of genes which are controlled to some degree, whose expression is controlled to some degree by genetic variation in cis. And excitatory neurons are the biggest cell population, and they also have a large amount of RNA typically. So we actually have very good transcriptome. We can map a lot better the QTLs at the cell type level. But we also went deeper at the cell subtype level and mapped the QTLs there as well. The main message I want to share with you here that we thought was quite interesting, and this is in a paper that's on bioRxiv, is that if you look at the QTLs that were mapped in the cell type level, which is shown in the color here, for example, for excitatory neurons in green, we have the green circle, and in gray are the ones that are mapped at the cell subtype level. So after we partition the excitatory neurons in the 14 constituent subtypes that we found. And what you can see here is that while the vast majority of them are shared between the two, that's of course expected. There's a large number of QTLs that are only seen here in green, the 1568, which are only seen at the cell type level. It makes sense since we have less power to find QTLs in the cell subtypes, which have fewer nuclei in each of the subtypes. But what's very interesting is this gray section here, the 1323 QTLs that we mapped only at the cell subtype level. So it really shows that mapping QTLs at the cell type level is insufficient. And there's, again, not unexpectedly, we found a lot of context-specific QTLs that are only emerging at the cell subtype level, despite substantial loss of statistical power to discover such effects. And this is not unique to excitatory neurons. We saw it in inhibitory neurons, and in every cell type that we looked at. And of course, we then use this information to map the effects of genetic variation from different disease GWAS studies. Here's the results shown for the Alzheimer loci. But also we can begin to look at very interesting context-specific effects that are providing mechanistic understanding for some of the disease-related traits. And what I've shown here on the right side is the expression of the APOE gene, of course, well known, a high risk factor for Alzheimer's disease and for other conditions. And we found a novel QTL that's only present in microglial cells. So despite also being highly expressed in astrocytes and being expressed in other cell types, we see that this QTL is unique to the microglia. what was very interesting is that you see here at the bottom on the right side, that the allele that leads to more ApoE expression in microglia is also associated with more cerebral amyloid angiopathy, so a form of amyloidosis that affects cerebral blood vessels. However, this variant does not affect amyloid proteinopathy in the parenchyma proper or tau proteinopathy. And all three of these traits are affected by ApoE4, which is mediated there by a coding variant, a pair of coding variants, and not the level of expression of ApoE. So we can begin to dissect actually the mechanistic effects of ApoE in different contexts in the brain. So now moving back to the main topic for today, which is looking at the properties of the cell subtypes in the neocortex in relationship to Alzheimer's disease. So the analysis that we performed, which is shown here in the middle in the discovery study, is to relate the frequency of each of the 92 cell types that we had discovered in our data to different traits. And the three traits here are shown in columns. So we have cortical amyloid pathology, cortical tangle pathology. These are the two constituent pathologies of Alzheimer's disease, and also the rate of cognitive decline that the person exhibited prior to being deceased. And you can see that the most significant result is a population of microglial cells, the microglial 13 subtype, whose frequency is associated with all three traits. And you can see a number of other cell types are also associated. Now we needed, of course, a replication, but we didn't have, a large number of single nuke data yet on an independent set of individuals. However, we did have bulk RNA-seq data from the same region, from the same collection of individuals, and that were independent from the ones that we had sequenced at a single cell level. About 408 individuals actually had both data sets. And so we used another method that Dr. Menon created, CellMod, to infer the proportions of the different subtypes in the bulk data, and then use this bulk predicted frequencies of each of the cell subtypes to confirm the results and ultimately do a meta-analysis of the two data sets to have more power. And that's just shown on the right. And you can see on the right side, again, the three traits, amyloid, tau, and cognitive decline. And again, with the same populations are coming up, microglial 13, astrocyte 10, for example. But now we have other ones that are also associated more strongly. Some shown in red are increasing frequency with disease, and in blue, some are being lost in frequency. Interestingly, again, and it's not just neurons, in the ones that are being lost, diminishing in frequency, for example, we see several oligodendrocyte species. This is just to show that the results are very consistent between the bulk predicted frequencies on the y-axis for each of the diagrams here, and the t-statistic from the single nuke data on the x-axis. We have amyloid on the left, tau on the right, and cognitive decline below. And again, you can see here that the results are quite consistent between the actual RNA single cell data and then the inferred frequencies. And again, microglial 13, which for the aficionados does have a high expression of the DAM2 signature, the disease-associated microglial signature found in mice, is the most highly significant population. Now, we also went beyond this to try to establish this causal chain. So where do these associated cell types fall in the sequence of events that leads to disease? Because we have data, antemortem and postmortem data, we can build statistical models using structural equation modeling. And again, I don't have time to get into the detail, but what we show here is taking the framework of the pathology, where amyloid comes first, leading to tau accumulation, and ultimately leading to cognitive decline. We place two of the most strongly associated populations, microglial 13 and astrocyte 10, in the sequence, showing that ApoE4, the strongest risk factor, which affects all three of these traits. But you can see here that ApoE4 affects A beta accumulation, which in turn then mediates a sort of the effect of some of the effect of E4 on tau, and then some of it is independent. Similarly, microglial 13 mediates an important fraction of the effect of amyloid beta on tau accumulation, but not all. So it's not uniquely mediating the effect of amyloid pathology on tau pathology, but an important fraction. And on the other hand, we propose astrocyte 10, the frequency of that cell type mediates a proportion of the effect of tau on cognitive decline. The main sort of symptomatic manifestation. So again, we're placing these subtypes of cells along the causal chain leading to Alzheimer's disease. Another way to try to infer trajectories is to plot the individual subjects. When here in this graph on the left, we are no longer looking at individual cells, but rather individuals. So these are the 424 individuals in our study. Each dot is one individual. And then we can try to color them the frequency of their cell subtypes. And I'll draw your attention at the top right to astrocyte subtype number 10 and 5. And you can see here, those two astrocyte subtypes are really defining the extremes of the distribution when we plot the individual subjects. Astrocyte 10 defining really the end, the terminus of the trajectory going south, and then this one going to the east is astrocyte 5 defined. And so we now have these two trajectories going to different termini, and this is very consistent with the heterogeneity of the older population in terms of who develops Alzheimer's disease when almost everybody is at risk for developing that disease. And very interestingly, the trajectory going south, going towards the bottom of the screen, is enriched for both pathology and cognitive decline. So really, this seems to be the trajectory towards Alzheimer's disease. Now, of course, what's happening to the people in blue here going with a trajectory going to the left? We don't know. And so there is, of course, some cognitive decline. There's some pathology there as well, but it's not clearly progression to Alzheimer's disease as in the trajectory. And so we call this alternative brain aging, and while the downward trajectory is the progression to Alzheimer's disease.
Philip, do you have many more slides?
Now, previously,
Do you have many more slides? Okay,
so,
great, thanks.
Yeah, sorry, I'll just wrap up. So basically, just to say here on the right side, that we also found correlations between different cell subtypes, which showed that the cell populations are not changing. Individual cells are not changed by themselves, but rather there are communities of different cell subtypes that are changing together, both in frequency and also in trajectory. And this is just to show that you can also see the association with one community that's enriched for Alzheimer's disease. So just to summarize, what I've shared with you are some of the functional consequences of AD variants that we were able to map with QTLs, the proposed causal chain that included some astrocyte and microglial cell subtypes, trying to address the heterogeneity of the older brain, trying to understand why some individuals may be more or less vulnerable to Alzheimer's disease. And I think the important insight also that the cellular communities in the brain may be the primary target and not individual cell populations. All these data are available through the AD Knowledge Portal supported by the National Institute of Aging, and there are 2 bioRxiv papers which delineate these results. Thank you very much, and I'll just highlight again Dr. Menon at Columbia with me, and Dr. Habib at Hebrew University, and my colleagues at Russian University, David Bennett and Julie Schneider, as well as Masashi Fujita at Columbia with me, who did a terrific job on these studies. Thank you.
Thank you very much, Philip. Thank you. I really like the mention of cellular communities at the end, but we have time for one question before we move on to the next speaker, if there are any. Okay.
If no one has one. Thanks, Phil. That was beautiful. Actually, my question is on T cells. Do you capture T cells in the profiling or not? And I ask that also because they're starting to make some, rounds in Alzheimer's now, and hard to profile.
Yes. Thank you. There's really a lot of interest in T cells and the adaptive immune component of Alzheimer's disease. In short, we didn't find enough T cells to really explore this question in a powerful manner. Amongst the 1.6 million transcriptomes that we analyzed, 90 of them, so 90, were for T cells. So basically, T cells are present, but really at a very, very small frequency. But we are now beginning to address this question using spatial transcriptomics, and we hope to make some progress there to see really what's going on with the adaptive component. Again, a very interesting question, but unfortunately, in this data set, we couldn't address it.
Okay, thank you very much, Philip. I'd like to welcome our next speaker, Neil Henderson from the University of Edinburgh, to talk about one of my favorite organs, the liver. Go ahead.
Thanks, Gary. Yeah, are there any other organs other than the liver? I'm not sure. People keep talking about them, but the brain of the abdomen or something. Okay, so yeah, first of all, thank you very much to the organisers for asking me to speak. I want to just put a shout out for our liver consortium. What a bunch of nice people you are to work with, and the Human Cell Atlas has brought us all together. And I'm not normally into group hugs because I'm a Scotsman and it's hard to talk about feelings, but genuinely, what a great consortium, and thank you HCA for bringing us together. Okay, so here's poor old Prometheus, and many Greek scholars in the audience will know all about Prometheus. So his eternal damnation was to get a bit of his liver eaten by an eagle or a vulture every night because he stole the flame or the fire from Pandora's box. And he's having a particularly bad day on this day because the eagle's got his claw in his eye as well. So he's really having a bad run. But what's, joking aside, what's important here is somehow the Greeks knew about liver regeneration, and how they knew that, we don't know, we can speculate, but they are smart people. We asked Prometheus if he was up for donating some liver to the Human Cell Atlas, and he said, that's not funny at all. Do you realise I'm in eternal damnation? So anyway, what we've done recently is we've been using multimodal techniques, and Aviv gave a very nice introduction indeed on all the various ways we can skin the cat now. We've used some of these techniques and technologies in this study, which we published in bioRxiv just towards the end of February, and if people are interested, please feel free to have a look at this paper. We've put all the spatial data, all the single nuke data into a web browser that's fully browsable. Okay, so acute liver failure, I just need to give you some background with this syndrome. This is a very scary syndrome, probably the most scary syndrome I look after as a liver specialist, because it's often out the blue. It could be someone in the audience next week, and I don't mean to be scaremongering, but you could develop acute liver failure from non-AT hepatitis. Thankfully, that is rare, but it happens. The commonest cause in the US and the UK is acetaminophen. We call it paracetamol, both accidental and non-accidental poisoning. And the problem is we don't have enough donor organs, so if we need to super urgently transplant someone for acute liver failure, someone on the chronic liver disease list doesn't get a liver and dies. So it's a big issue. So us and many others are interested in looking at how we can develop pro-regenerative therapies, because we know the liver is intrinsically regenerative. It's how we harness it and coax it to become even more regenerative. So I'd like to just introduce some histology here. Well, not quite histology, but schematics. So the liver has a beautiful 3D repeating architecture, and this is a liver lobule on the left, unhealthy, and we have the portal vein going to the central vein across the liver lobule. When patients have taken either accidentally or non-accidentally acetaminophen in too big a dose, they wipe out the hepatocytes in zone 3, the so-called pericentral vein region. So we're left with the PNR, the perinecrotic region, and the RVR, the remnant viable region. Now, ourselves in my group and many groups have basically thought that the way the liver fills in these potholes of dead hepatocytes in this type of setting is by proliferating hepatocytes, and that's what fills in the hole, and that's been the dogma for as long as I can remember. So what we sought to do, and we didn't necessarily think we would get into anti-dogma type stuff, but we set out to make a pan-lineage atlas of human liver regeneration using single nuke-seq. So we reached out to friends all around the world who had biobanks of acute liver failure tissue, which is rarer, much rarer than chronic liver disease, and we generated this atlas. And so I'm just going to go, in the interest of time, I'm going to dive straight for the hepatocytes. This is the major epithelial cell type in the liver. And what struck us once we'd done the initial clustering was this cluster of ANCSA2 positive hepatocytes, which was one of the differential marker genes for these hepatocytes. And you can see that they're very expanded in both acetaminophen acute liver failure and non-ATE hepatitis, the other big cause of acute liver failure. And when we did geoanalysis on those specific hepatocytes, the migratory terms, we're absolutely leaping out the geoanalysis. And we all know you have to take geoanalysis at times with a pinch of salt, but every time we looked at it with different types of geo approaches, it was epithelial cell migration, amoeboid movement were at the top every time. So this intrigued us because hepatocytes aren't thought to be very migratory. Normally we think they proliferate, but they don't move much. So this caught our eye and we moved forward with this. So Kylie in the lab, who's the wet lab postdoc leading on this project, stained for ANXA2, and we could find ANXA2 positive hepatocytes populating the edge of the necrotic region, and they're enriched in acute liver failure. And then when we looked, because we've been running the transplant unit in Edinburgh for about 35 years now, we've transplanted for lots of different etiologies of acute liver failure. And you can see here hepatitis A and B on the top, ecstasy bottom right. We've unfortunately had to transplant 3 or 4 people for ecstasy-induced acute liver failure, and terfenidine's an example of a drug-induced liver injury. And what we saw was a motile morphology to these ANXA2 positive hepatocytes, irrespective of the cause of acute liver failure in humans. So it looks like a common response to any type of human acute liver failure, certainly all the ones we've screened. So I'm now going to flip from human to mouse. I often, not in a negative way, but will criticise some of our mouse liver models. For example, non-alcoholic fatty liver disease. We're trying to recapitulate a disease that might take 20 years to develop in humans in 6 weeks in a mouse, and that's a challenge. This is actually one of our better mouse models, the acetaminophen-induced acute liver injury model, because it recapitulates a lot of what we see in humans and is actually really quite a helpful model in mice. So Kylie did a fairly heroic time course top left, where she gave acetaminophen in cohorts of black 6 mice and then took liver tissue at multiple hours post acute liver injury, again generating an atlas like the human atlas. And then what we did was we took the human hepatocyte subpopulation, ANXA2 positive subpopulation, and that off that we could generate a migratory signature and overlaid it on the mouse data. And clearly the beauty of the mouse model is you can go longitudinal and start to look at when this migratory population starts to become overt and more manifest. And you can see in the tail of the fish, if you like, we could start to pick out migratory hepatocytes in the mouse model. I'm just going to labour this slide just for a second or two, because we think it's important in terms of we wanted to pull apart whether indeed it is hepatocyte proliferation that is healing the necroinflammatory wound or the pothole around the central vein. And so again, we did a fairly forensic time course where Kylie took tissue at all the hours along the bottom axis. And then what we started to tease out was that actually about 50 to 60% of necrotic wound closure, so filling in the pothole, occurs before hepatocyte proliferation in the green colour even gets going. And so that was quite striking for us in terms of we'd assumed that it would be hepatocyte proliferation that would repair all the damage, but actually more than half of your wound closure is occurring before hepatocyte proliferation even gets going. So that's interesting, and NucSeq gave us a lead into that, and then all the other mouse model work, but we would love to try and see these migratory hepatocytes to see if we can actually see any evidence of that. So this is a mouse where we can turn all the hepatocytes red with a TD tomato reporter listed on the bottom with a viral induction system with a TBG promoter, which is hepatocyte specific, and a multiphoton setup that I have a friend in Glasgow who does a lot of lung multiphoton, and he's a really great collaborator, and he was happy to go south to the liver, so we started imaging the liver. And what you can see here is when we induce acetaminophen injury in mice, this is just going to run on a loop, but you can see the zone 3 necrosis in black, and then wound closure of the hepatocytes between 36 and 42 hours after paracetamol. We chose that time point because all our longitudinal data suggested that was when these hepatocytes were becoming manifest and expressing ANXA2. If you zoom in, you can start to get a handle on how the closure is actually happening. I'll hopefully draw your eye with arrowheads to the hepatocytes at the edge of the wound start to develop lamellipodiae, they start to ruffle their membranes, they start to move. And what's striking is it's almost always hepatocytes just at the leading edge. We don't see this morphology, if you like, in the rows further back. And again, last video here, but just to show you, you get increased nuclear rotation, which is commensurate with cells that are migratory. And again, this reaching out and stretching of the membranes and ruffling. So then, using a different type of reporter mouse, this is the MTMG mouse, so when you express Cre in hepatocytes, it goes from membranous tomato to membranous GFP. And the beauty of this mouse in this setting is you can then segment every individual hepatocyte in your intravital videos. So Kylie and Fred in Leo's lab, who's an expert in quantitation of intravital microscopy, then got stuck into this in terms of working out exactly which hepatocytes were moving on a cell-by-cell basis. And this is just some of the data, but you can look at things like cell mobility with the various parameters you can see there, speed, displacement, etc. And the take-home message here is that the hepatocytes in the perinecrotic region moved much more than what we saw in the rows further back, commensurate with this kind of pulling in and closure of wound. Then knocking out ANGSA2 specifically on hepatocytes with a viral system, hepatocytes are very tropic for adeno-associated virus 8, so you can give them short RNAs, and it's a system that's used a lot in liver. So we knocked out ANGSA2 on hepatocytes. In essence, after paracetamol, you can inhibit wound closure by knocking out ANGSA2 on these hepatocytes. Now, how ANGSA2 is mediating the wound closure, we're in the thick of trying to work out. We can speculate at this point, but we're currently trying to tease apart mechanism of how ANGSA2 regulates that on hepatocytes. So in essence, what we've got towards is an overall model where, as I mentioned prior, we thought it was hepatocyte proliferation driving this wound closure, but what we think is happening temporally is that you have peak necrosis with a wipeout around zone 3 of hepatocytes dying in that area. You then have this wavefront of hepatocytes that are moving. I think it is genuinely very similar to skin wound closure. So if we have a cut, we want to close that wound as fast as possible because it's a pathogen barrier. And we think similar is happening in the liver, where you're getting a lot of wound closure before you even kick the hepatocytes to proliferate. The other thing we know in acute liver failure and in chronic liver disease is the gut is very leaky. And so the liver's seeing more antigen, more bacteria from the gut. And so you have this breakdown both in the gut barrier, but also in the liver of the gut-liver barrier. And in patients who we can't transplant because we can't get a liver fast enough, 95% of patients die of sepsis and multi-organ failure. So what we're trying to think about here is whether we can guide some therapeutics towards more rapid wound closure and restoration of the gut-liver barrier rather than driving hepatocyte proliferation per se. And I didn't show the human data, but actually in human explant livers, the rates of proliferation are often 60, 70, 80%. So hepatocytes are very good at proliferating. I think perhaps we need to think more about how we can close this wound faster in these patients. So I'm just going to end there and acknowledge Kylie, John, Jordan, and Andreas, who Kylie's a wet lab senior postdoc. John, Jordan, and Andreas are bioinformaticians in our group, and they did a huge amount of work in this project. I'd like to acknowledge the patients, the transplant coordinators, and all the surgeons who kindly helped us get tissue for this project, and our collaborators around the world. In particular, the US acute liver failure group were very helpful and sent us frozen tissue, as well as Cambridge and Birmingham in the UK. I've mentioned here some web browsers for our liver single cell data sets, both chronic liver disease and also the liver regeneration data. Sorry, that's the opposite of a pithy, concise web browser there, but it's in the bioRxiv paper, so please click on that. And finally, to the Human Cell Atlas, CZI, and the funders for funding this work, and thank you for your attention.
Great, thank you, Neil. That was amazing. So we have time for one question from the audience on virtual or in person. Any questions? Put up your hand, yes?
Hi, Neil. Great job. Really nice story. I don't know if you said it and I missed it, but in terms of proliferation, so do you think there is a base, if you imagine the column of the closing the wound, a base of hepatocytes that proliferate, and then at the tip, like in angiogenesis, you got the ones closing in and migrating?
Yeah, no, I know exactly what you mean. So I'm going to enjoy completely speculating in an evidence-free zone from here. But so when you look at, and I'll keep it brief because I know we're tight for time, but if you look at hepatocyte proliferation in this setting, it is commonly in the rest of the viable lobule. it is commonly not that much at the leading edge. So what we wonder is, when you get stretch and wound closure without proliferation in about 60%, we wonder that there is then a stretch and a mechanotransduction type pull on the rest of the epithelial sheet that then can potentially trigger hepatocytes into proliferation in the back rows, because you've not generated any new hepatocytes at this point, so there must be stretch on that sheet. So we wonder that that's what then triggers, if you like, the back rows then proliferating.
Yeah, so it would be looking at time courses after the wound is closed. Yeah, that's
Exactly.
cool. Really great. Thanks.
Thanks.
Thank you. So we're going to move on in the interest of time to make sure we get enough break with our next speaker, Tuuli Lappalainen from the New York Genome Center and Columbia University. Welcome.
Hi, thanks for the introduction. Thanks for having me here and sharing some thoughts, lessons learned regarding understanding genetic variation in the human genome and its impact on the transcriptome and other molecular and cellular functions. And obviously the problem that we're dealing with is the variant to function problem, or V2F difference, which is now emerging as probably one of the biggest challenges in human genetics, where there is this tremendous task of trying to understand what are the molecular and cellular effects of genetic variants that mediate GWAS and other links between teeny tiny genetic variations in human populations, and then diseases and traits that are extremely complex. And this is relevant for understanding mechanisms of disease associations and potentially developing interventions against those. My lab works a lot on sort of functional genetic architecture of human traits, very sort of generalizable questions about how this whole thing actually comes together. And there's also applications in molecular precision medicine. But today, instead of sort of jamming a couple of specific scientific stories into these 12 minutes, I thought that I would actually kind of give a bit of a general overview of what's been going on in this field and what are some of the sort of lessons learned and challenges arising that are particularly relevant to this community. So this is actually my first Human Cell Atlas meeting, and none of the things that I'm talking about here uses Human Cell Atlas data, but I think that there is interesting sort of like interfaces that we can maybe discuss over the next couple of days. So this variant to function question is not actually sort of like a single question. There's multiple layers of questions about causal variants versus LD proxies. There are sort of effects on, let's say, transcription factor binding, RNA protein binding, enhancer function, target genes in the locus, and then effects on cellular programs and regulatory pathways downstream that then ultimately link to physiological phenotypes that we can capture by GWAS. And then obviously here, there's the cell types and cell states where these causal processes are taking place is an absolutely crucial question. But there's a lot of questions to answer. Luckily, there is also multiple different approaches that we can take to tackle these questions. The one where I have done most of my work during the past 10, 15 years is in the space of molecular QTL mapping, where we leverage variation in human populations. Phil already talked about this, basically linking genetic variation to molecular and cellular traits. Gene expression, EQTLs has been the most popular molecular trait to look at, but this approach extends to all kinds of molecular traits. Then there is the slightly more novel approach where we also work, which is using CRISPR and others on large-scale perturbations of the genome and high-throughput readouts to analyze how perturbations of the genome affect molecular and cellular traits. And then there is the sort of the third approach, which is kind of sort of doing inference of variant function without genome perturbation. This is the sort of approaches relying on ENCODE data and sophisticated computational models on top of that to basically infer what variant functions might be. And when it comes to the molecular QTL mapping, I've been fortunate to be part of many large-scale efforts over the past more than 10 years, I guess now, to integrate genetic variation and typically the bulk transcriptome, sometimes other molecular phenotypes, sometimes a little bit of cell type information. But this, as you know, the scale of single cell data in this space has been relatively limited thus far due to just cost and practical constraints. But what has happened in this space is really creating data from hundreds and thousands of human individuals, nowadays tens of thousands, and linking those variants to molecular changes across multiple different tissues. Here, the biggest project in this space is the Genotype Tissue Expression, or GTEx project, which is now continuing in the form of developmental GTEx. Another approach that my lab has been pursuing, in particular in collaboration with Nevels and Jana, has been using CRISPR perturbations, in particular enhancer silencing or enhancer activation, to characterize gene expression effects of GWAS loci, using basically cancer cell lines or other sort of models that we can have on the dish. And here's some recent work from this space. And some of the local folks here may recognize the first author here. John Morris is a shared postdoc in our lab and will be starting his own lab in Toronto next spring. But it is very exciting that these types of approaches are also now producing very large-scale data about transcriptome and other molecular and cellular effects of GWAS variants. The reason why my lab has been very interested in sort of like pursuing these both approaches at the same time is that both of these approaches have benefits and caveats that are luckily largely orthogonal. And in the best possible case, we would be able to sort of distill into those advantages of each approach, such as being able to actually capture primary cells in their somewhat native tissue context by analyzing tissue samples collected from human individuals. And then, of course, when you do perturbation experiments in vitro, you have that kind of clean experimental models that is sometimes easier to interpret. And I very much believe that the sort of integration of the two is going to be important for our field. But so in the next couple of slides, I want to highlight some of the lessons learned from this type of work that has been going on by us and many others in the community. And this is sort of like a cherry-picked set of lessons learned. There is a very long list that I could have highlighted here. But some of the things that I think is that are relevant is, well, first of all, something that is very much preaching to the choir here: cell type composition in bulk tissues is a major complicating factor in bulk tissue molecular QTL studies. So we see these effects in many different forms. One of them is that merging independent QTLs that are active in different tissues when you analyze a complex bulk tissue sample basically sometimes leads to sort of like a one big mess of an association signal. And having that cell type resolution helps you to actually get additional resolution into the genetic effects that are taking place here. There. We can also use some statistical methods here, but obviously having just better data that doesn't clump a lot of things into one is also helpful. We have also seen that these sort of gene by environment effects at the molecular level, that has been one of the great areas of interest in human genetics, is very much complicated by changes in cell type composition. So here we are thinking about sort of functional effects of genetic variants that are somehow dependent on or interacting with the physiological factors, phenotype, environment, etc. But we can see that, and this has been formally shown by mediation models, is that these types of interactions that we capture in molecular QTL studies are often due to differences in cell type composition. So, for example, in this work that we recently put into bioRxiv, we can see that there is a general association between age and percentage of neutrophils in your blood, and then we also have obviously neutrophil specific, in this case, methylation QTLs, and what comes out of the combination of these two is a lot of loci that look like there is an interaction between that the effect of this variant on methylation levels is dependent on age, when it's actually just basically a mediation effect by neutrophils. And this is a complicating factor in all of these kinds of studies. And finally, just to briefly mention, when we are interested in genetic variants that have effects on gene regulatory programs, not just in the cis locus itself, but further away, these are greatly complicated by variants that affect cell type composition and then arise as kind of like false positives. The second point that I want to raise is that even though a lot of our work, and I think your work, is often focused on gene expression levels, splicing is now recognized as a major molecular mechanism for GWAS loci. So we have seen in GTEx data, and there's many other papers as well, showing that when we look at the sort of enrichment of GWAS signal in different types of molecular QTLs, the enrichment that we have for splicing QTLs is almost as high as for eQTLs. And we also know from a lot of work that the eQTLs and splicing QTL signals, those variants are typically distinct. So you will not capture those splicing effects by looking at expression alone, and splicing is really important. So let's not forget that, especially now that we're having better and better methods for actually being able to capture splicing also at the single cell level. Thirdly, this is work that is ongoing in my lab and kind of coming out of the work that we have done with John and with Neville, looking at this CRISPR-I of GWAS enhancers and looking at their expression effect in K562 cell line, and then looking at eQTL data in different blood cell types from eQTL catalog, GTEx, etc. Is that the overlap of the regulatory effects that we see in these, looking at the same loci, is really not great. There are, like most of the time we may see an effect in one, but it's missing in the other, pointing in the different direction. And we don't actually know why. This is something that we're digging into now. There is obviously problems that potential false positives in either approach, potential very high degree of cell type specificity, whether enhancer silencing actually captures SNP effects. But this is one of those things that we, I think, really need to figure out as a community when we have two major approaches and they give different answers. We really need to figure out how do we use a combination of the two in a way that actually leads us to whatever a correct answer would be. And finally, I want to highlight that sample size that we have when we do molecular QTL mapping, when we look at how genetic variants affect molecular phenotypes, is that sample size in these studies really affects the biology that you are able to see. And if there's one lesson that we can kind of like learn from the whole sort of history of human genetics and GWAS, is that increasing sample size has really had a transformative effect on biological discovery and applications. Early GWASs weren't really that impressive, and now the whole field has changed just by kind of something that sounds a little bit incremental, just increasing sample size, but it has really had an effect. And molecular QTL mapping is something that you can do with just, let's say you have 60 samples or 80 samples, you can run these things and you will find some effects, but you will often miss all kinds of more subtle effects that are more context specific, that are sort of affecting not just sort of genes proximal to these variants in cis, but also cellular programs in trans. And these are all properties that we know are particularly important for GWAS variants. So basically, small-scale molecular QTL studies will often miss the most important things. And here, having very deep molecular data is great. Having very nice methods for analyzing that data is great, but when it comes to genetic association studies, there is no way to replace the number of individuals and the statistical power that you get there. So here, we really kind of like need to push the boundaries here and pursue large sample sizes. So thinking about going forward, I think this is, again, preaching to the choir. Having molecular QTLs with cell type resolution, this often means single cell resolution, is, I think, extremely important for the field to tackle some of the remaining challenges and really push the field forward. This will help us to integrate these data with GWAS and the entire work that is going on in the sort of human genetics field. Here, immune cells are an obvious place to start, and that this work is ongoing. But obviously, blood and PBMCs and immune cells are in the whole picture. We need to pursue these studies across multiple different tissues, incorporating ancestral, environmental, phenotypic, developmental diversity into these studies, and then also thinking about the multi-omic aspect that captures effects on splicing, chromatin state, etc. It's already been highlighted by Aviv and others that there is kind of increasing attention to cellular programs. A lot of the variant to function field has been quite focused on sort of identifying the causal gene in the cis locus itself, but getting to cellular programs is extremely informative and important. And finally, this kind of integration of genetic approaches and perturbation approaches and sophistication in how we analyze and produce this data is great. I'll stop there with quick acknowledgements, and I thank you.
Thank you very much. So again, in the interest of time, to give us a break, we're going to take one question. So I think Muz wanted to do one before, so couldn't do one, so I'm going to give it to her.
Thank you, Tuuli. Muz Hanifa here. That was great talk. Love the lessons. Very quickly alluding to the kind of cellular programs, and I guess we're all a constellation of variants, and it's always been looking at one variant to sort of function, and I just wondered if you could elaborate a little bit more what your views on how we can take a kind of more sort of like more biologically relevant perspective to how variants affect function.
Yeah, so I think there is many potential answers to your question, but maybe one that I can give is that I think that is actually, when we think about cellular programs, that is a place where integrating this sort of polygenic thinking about how genetic variants affect cellular programs, cellular phenotypes, is actually not conceptually that difficult to integrate. And actually in the recent science paper, we were able to show that there is this sort of GWAS enrichments in networks that we can incorporate. So basically tackling the variant to function problem, not just by kind of analyzing locus by locus and looking at what is this variant's effect on potential regulatory pathways, but actually analyzing overall enrichments of GWAS signals in those pathways works reasonably well. But it's promising. Yeah.
Great. I heard we had time for one virtual question, so.
Fantastic. This is from Shyam, Prabhakar. I'm curious to know your take on MEQTLs versus CQTLs versus EQTLs. Under resource constraint, which would you prioritize?
Yeah, hi Shyam. Great question. I don't think there is a single answer to that question, and it's also like very much of a question of let's say, like what works, which assays work nicely in the tissue samples that you have. I'm still a big fan of the transcriptome in the sense that, I mean, it is kind of like the first molecular phenotype that kind of leaves the genome. Chromatin marks are super, super informative, but it's still kind of like there at the genome regulation level. And I think that they also answer a little bit different questions. So which one is your priority depends on what are you actually wanting to discover and do with the data. So sorry, I'm not going to give a straight answer.
Okay, thank you very much. It'll be interesting to hear how variation plays into all of the different topics that we will talk about for the rest of the meeting. So thank you to all the speakers. We now have a break until 10:55 as originally scheduled, so it's about 16 or 17 minutes, so please make use of the break time, and we'll see you back here in the main hall at 10:55.
Okay, so let's get going with the next session then. So the session here is going to be a series of really building upon the introduction that you heard from Aviv and the other speakers in the first session, talking about some of the scientific highlights, work on integration, a real focus on how the inclusion of diverse populations is important for assembling a meaningful atlas. And then we'll close the session with a discussion that focuses on really the key theme of this meeting, how to define the first alpha version of the human cell atlas, and equally important, how that atlas is going to be used. So to kick off, it's a pleasure to introduce Malte Luecken from Helmholtz Munich, who's going to give an overview of some of the efforts for Atlas integration that have taken place over the last year. So Malte, over to you. Where's Malte? There he is. Excellent.
Thank you very much for the introduction and the kind invitation to speak today. My name is Malte Luecken. I'm a PI at Helmholtz Munich, and I also coordinate the Human Cell Atlas integration team, where we're working together with bionetworks to build reference pilots for all human cell atlas tissues and organs. If you're interested in anything I have to say, please just catch me at the event, or feel free to get in touch via various forms of email or social media. As I'm sure you're all aware, the last, going on approximately a decade now, has seen many single-cell data sets of the same tissues and organs being generated. This is an example of several recent studies from the lung, and Aviv showed 128, I think it was, this morning across different tissues and organs. So it may come as no surprise that each of these studies will show a different view of what the healthy organ looks like, but they will also disagree in their cell type nomenclature, in which populations are studied, etc. So if I want to ask where is my favorite gene, maybe a viral entry factor expressed in which cell type, how do I do this across all of these data sets and taking into account the diversity of the populations that have been profiled in each of them? This is something we are trying to tackle with data integration or removal of batch effects. If I take a lot of studies, process them in a similar way, typically these studies will be organized, the cells within those studies will be organized by their batches from which the cells come rather than the cell types. Data integration is trying to remove, or batch correction is trying to remove these effects that enable technical factors to dominate the representation of cells and instead focus on biological meaningful results. Now, of course, if I apply different approaches, I will get different answers. And here is just 3 different outcomes shown in a quick UMAP representation of a couple of the lung studies put together, where we could get very different conclusions from looking at these representations. So in order to not have to go through all of the approximately 200 integration methods that have since been published or are available at the moment, we, in the past years, developed kind of a pipeline to quantify what integration success means from 14 different aspects using 14 different metrics, both including the removal of batch effects as well as the conservation of cellular variation, whether on cell type level or other. And what that looks like for taking studies from the lung is that we get a ranking over here from top performing methods to less well performing methods on the basis of these different metrics. And that was made available as a pipeline a couple years earlier last year, which can be used to kind of test which information methods work well for different scenarios and really give you different solutions depending on what works best for this particular case. Lisa, a very talented PhD student in the Teichmann lab, spearheaded the project and to use some of these resources and the studies that are available in lung and take consistently curated data and metadata, compare integration methods to see what works best for the human lung in this case, and come up with a core atlas, which at first glance looked great. We could separate kind of broad cell type populations. However, we quickly noticed that this is not something which we are particularly amazing experts in. And so this is a project which really was done together with the Human Cell Atlas Lung Biological Network. And we asked experts across the network, and here in this case 6 different experts, to annotate each of these clusters which we find to come to a consensus. And in this consensus, each cell type or cellular identity here is represented from cells from multiple samples, multiple data sets. And so therefore it's not kind of a one opinion or one single outlier. We believe that these are really supported widely. It turns out it's not only a tool once we have this to it's not only a powerful tool to generate cell type consensus, but once we have this reference, we could discover rare cellular identities, which were not purported in some cases in the adult human lung. We can associate genomic variants with cell types using kind of a larger resource that we have. We can look at the effects of age, sex, smoking status in various genetic pathways on cell types and gene modules. And potentially the most important or most transformative application which we've found since then is that we can rapidly analyze new data sets that are projected to this reference to project labels. And since this has been made available, the reference has been downloaded over 1,700 times. And we also made this available at the time on a platform when there's been approximately 70 users, which is registered there to kind of produce 100 different mappings. Now, given the appetite clearly for a human lung cell atlas, there are many more different opportunities for different human cell atlas bionetworks who are looking to answer different questions, but also looking to kind of generate references, as kind of Aviv talked about this morning, as the goal of the Human Cell Atlas Consortium as a whole. And so we took what was done for the Human Lung Cell Atlas and tried to identify broad steps that we would need to kind of use to address these quite different problems, but in a consistent manner to have common quality standards. And within the Human Cell Atlas integration team, we're doing that. We're focusing on broadly common data curation, method selection in a principled manner, annotation in a community, across the community, and then use in very different uses through different problems. And as a first phase, we'd focused on 6 pilot projects, which I'd like to tell you a little bit more about today. And the second phase, which is starting in a couple days, where there are 6 more projects which we are initiating, and then looking for a stage 3 to look kind of at how we go across those potentially and address, to work with the rest of the human cell atlas bionetworks to build initial reference atlases. The difference from why this is not just a computational integration effort, but this is really a human cell atlas referencing effort, is the input and the crucial collaboration with the human cell atlas bionetworks on the data curation side and, very importantly, on the annotation side. As I mentioned before, as a computational biologist, I am not the expert on any of these organs. The experts are in this room and in the virtual participation in the wider world, who each have their own expertise and opinion on different cell types. And this is the conversation we really need to be having on the basis of data which we're trying to put together within this team and in collaboration. So this is not me doing this. I need to introduce the team very briefly. So here you see pictures of and in bold of the team, current and former members, as well as counterparts in the human cell atlas biological networks who are working in these different projects to really make them happen across kind of brain, kidney, gut, or small and large intestine, version 2 of the lung, the retina, and peripheral blood. And of course, these integration projects are all quite different. Each organ has its own challenges, and therefore we need to tailor solutions to make sure that these challenges are addressed. And I just want to quickly give you an overview of what some of those challenges are, which mean that we need to tailor these solutions. I'm not going to go through all of this, but just to give you an idea, kidney, for example, we found has very, very strong batch effects from dissociation and sample handling. So as a solution, we're trying here, Peng and Konstantinos, Konstantinos is here today as well, is testing if we can realign all of the data just to make sure we remove that batch effect a little bit to ease that integration burden. In the retina, we're doing cross-species integration because annotations from macaque and mouse, in some case, really can help us to understand how these cell clusters, these cell identities in the human maybe should be called, or just give a hint. As well as single cell, single nucleus integration, which sometimes does and doesn't work across different projects, et cetera, and mapping multimodal data. Nonetheless, despite all these very diverse challenges and tailored solutions, we want to set up consistent standards for the quality of an integrated reference. And these come from working together with each bionetwork and trying to learn across those bionetwork projects for common quality standards. The approach we have in phase 1, we're really doing kind of using the data ecosystem as well to leverage kind of consistent curation of data sets with the bionetworks and metadata processing after defining an atlas scope. Then we go into looking at individual data sets to run the QC there. In a second phase, we really try to prepare a comparison of integration methods. And this comparison is tailored to the particular questions that each project has. Here, we're trying to get a consistent label encoding so that we can computationally compare these methods and then do some further validation work. And then in a third phase, we perform the data integration, cluster, and re-annotate this in community sourced jamborees. And then there's this downstream analysis, which I showed there, which is a whole host of things which will be very different, and therefore I'm just calling this downstream analysis for each project. As mentioned, this integration benchmark is really the crux here, where we want to compare different methods to see that we can tailor the approaches best to the questions at hand. And here in different projects, we're asking questions of, is it better to integrate per lineage? Is it better to integrate single cell and single nucleus together or as separate representations? What if we integrate disease with healthy data? And do we need to realign? The current pilot projects are at different stages of this pipeline. This is now approximately, depending on the oldest part, maybe 9 to 10 months in. And a lot of, because I can't talk to you about all of these different projects in the very limited time I have, I want to focus on an aspect where a lot of these projects are currently, these projects are currently working on this second technical phase. And please, if you're interested in this kind of general this general kind of standardization and QC pipeline, listen to Michaela's presentation later today in the kind of breakout session on integration pipelines. Yes, to get back to the part where quite a lot of these projects are currently working on. To set up a comparison of integration methods, we need to have a computational encoding of cell type information to capture the author intuition or the data generator's intuition in a way that we can compare these integration methods computationally. This is not the same problem as re-annotating the cells. We do not need to know exactly what the meaning of different labels are. We just need a coarse kind of agreement between these different studies to say that, I understand if I'm mapping these two together, I'm not doing anything terribly incorrectly. And this will help us to understand how different integration methods are performing. So for example, if I have 3 different data sets with very different annotations, what I'd like to find is some kind of alignment. This can be 1 to many, many to 1, of an intuition of where these labels should be mapping. And there are many different methods that have been specifically designed for this, or in some cases that we're trying to use within the team to tackle this problem. Chuan has developed, who's also in the integration team, has developed a cell typist harmonized approach, which tries to computationally align. We have been testing Graham's, who's also here today, similarity approach, and he's also going to talk about this in the session I mentioned. The cell annotation platform is kind of meant to be the canonical way to address this by coming together with all these individual data sets. And then, of course, we can just do this manually. I can't really talk about this more, but if you're interested in this, there's a poster from the blood integration team, Michaela and Anna Maria, who have compared a couple of these different methods. What I do want to mention briefly is that we found that we can maybe speed up this process a little bit when there is a previous reference already in place. In the second version of the Human Lung Cell Atlas, and incidentally also building a gut integration, we find there's quite a decent, a pretty good first version of an integration. We can map data sets onto a reference and thereby learn from this reference what maybe the labels that are consistently encoded could be. What that looks like is if I take a new study and I have 2 cell types annotated as cell type 1 and cell type 2. I don't know what cell type 1 or cell type 2 are. I want to keep the author intuition now, but ensure some kind of consistent encoding. I can apply this reference to predict cell type labels, and that's not going to work perfectly at all, right? And now suddenly I have some unknown labels, A, B, and C. But I take, again, the author's original intuition by using the clustering that the author did, that the data analyst did, and then look at a majority vote in there. And now I have this A and B relabeling, which are the same cell types. And I can do this with every single data set to try to get a consistent encoding. In practice, this is a little bit more complicated because we also have hierarchy in these labels. But we found in the lung, for example, that comparing this to manual label transfer, this works quite well. In some cases, we could give more resolution, which we don't really want to do because we want to keep the original author's intuition entirely when we go to level 4 and 5. But in general, this works quite well. But of course, we don't want to trust this automated process fully. And so in all cases, we then look for markers that have been reported in the reference either or have been uploaded to the cell annotation platform that we can use to assess, yes, okay, these labels that we are encoding in a consistent manner are the same across the board. And that really then sets us up to do this benchmark, to run this benchmark in a different manner for different projects. What I want to, a reason I'm showing you this briefly is that this is really a prototype for a fast reference versioning. So here we are building a lung version 2 in a much faster way because we can learn from lung version 1 and this complicated process of like consistent encoding, which has been taking months to do in the pilot projects, we can do in very, very fast iteration. And thereby, we can go to map, re-benchmark, re-annotate with community input in a way that's much, much faster than doing this from scratch every time. Thereby kind of paving a way for future versions of these references because version 1s, as we've also mentioned, are MVPs. They are not going to be perfect. We've identified several limitations with the one we already have, and we're trying to address those now going forward. To summarize, I want to leave you with some pretty pictures because I got a lot of slides from the integration team, and I think they're really cool. I couldn't talk about too many of them. But I want to tell you that kind of what we're trying to do is build kind of community consensus on the basis of a common data set, which we're building together with the community who is we were looking for input on for this consensus. We're trying to adapt the lung cookbook in a way to tissue-specific challenges, expanding it and really making this a very specific project. And in this doing so, we still want to achieve common quality standards. Therefore, we have this kind of quality control and standardization pipeline, which is not to say that these are it's a standard approach for everything, but we really want to make sure that at least we've done similar vetting of this. And the next steps, benchmark runs and annotation jamborees. So if you're inclined, please, in the next couple months till the end of the year, we're looking to have run a few, up to 6 of these annotation jamborees. So, and I then need to thank a lot of people who have done a lot of work on this. The integration team, the bionetwork counterparts, the senior advisors of the integration team and support, as well as the bionetwork seniors. And there's 3 dots at the end of the HLCA because this is, I couldn't fit all the names on. Usually I have another slide of annotation, of acknowledgements. This is really a community effort. I'm just standing here telling you about it. Thank you.
Thank you, Malte. Any questions? When there.
Thank you. Awesome presentation. Just wondering, when you go to integration and handling things like batch effects, how do you control for things such as the quality of the tissue, moving from, say, a healthy donor to a deceased donor to an inflamed setting? How do you approach thinking about whether these batch effects are actually removing some important biological information that would tell us something meaningful?
Yeah, so this is a really good question. And the thing is that this question is something you kind of have to answer differently depending on the tissue context as well. Where you might not have, I know from Chloe, and I'm sure she's happy to talk about this, that in blood, that the time after taking the sample, how long is it standing on the desk is going to be really important. That's maybe not something that's as relevant. There's been a comparison in lung, spleen, I think, from the Teichmann lab, that up to a certain time it doesn't have as much of an effect. And so it's difficult to give a general answer to that. The way we're approaching this from the starting point is that we have several aspects, summary statistics of data, which we're trying to calculate to say, is there any outlier in these data sets for a particular tissue? Because we can't really tell you the exact range of what is good or what is not good, but if it's an outlier, we should pay attention to that. And that's how we start trying to focus on the individual data sets, which we might have to pay more attention to. And then try to take as much bionetwork intuition as possible to test for other things like inflammation of proximal tumor tissue, for example, which might exactly be a situation that you mentioned.
Another question.
Yeah, Matthias Kassler, comment to that. Particularly for the network starting in a couple of days, what's exceedingly helpful if you can establish a set of reference samples, meaning provided from one single source, which you can distribute to your units generating data. In the kidney, we were fortunate enough to do that with a standard analysis working group in a human cell, with the kidney seed network, with HubMap, and with KPMP. So you have at least one benchmarking available that the same tissue has been run on different platforms, and some of the pre-analytical steps therefore can be integrated. So think creatively, if you're starting a new effort, is there a common reference source which you can include going forward?
So this is exactly why we're working with Matthias on this kidney project, because we get the data downstream. I can't tell you why the data is good quality or not. I think that's a really good suggestion.
So question at the back, and please introduce yourself when you ask the question.
Hi, I'm Hari Nakshatri from Indiana University. Our breast studies are done with the healthy donor's breast tissue. They are collected and stored within 6 minutes. Whereas many other groups have published the single cell atlas of the breast using reduction mammoplasty samples. They are collected during surgery, which takes longer time. So there are already sets of data publicly available where one is collected through a standard operating procedure with only 6 minutes exposure and others from the surgical samples. So there may be precedent where you can compare those two atlases data generated to come up with something what the tissue collection procedure itself has an influence.
I see. We're currently not as much working on kind of comparing data generation approaches, also because to do that, we first need to have a reference in the first place, and we cannot necessarily be choosy. Because we need to, I think there's two aspects. We need to be kind of as broadly encompassing as possible for different people's expertise, which is put into their data samples, right? And secondly, it's great, there are a lot of data sets, but if we look at things that match our quality criteria in the end, it's still kind of a growing procedure. So I think using as many data sets which do match the quality criteria we set at the end is very helpful. If in some cases we could do better by removing some, that might turn out to be the case. Yeah.
Thank you. I think we'll have to move on to the next presentation in the interest of time. Thank you, Malte. Let's thank Malte again. And the next speaker is Ed Lein from the Allen Institute. Ed.
Thanks, John, and thank you very much to the organizers for the opportunity to tell you a bit about progress in brain today. The theme I'm going to be talking about is sort of moving from pilot atlases that we've been creating for the last number of years to really comprehensive atlases in human, and then moving from this sort of draft version to community standards. In many ways, I'm going to be representing the work of the NIH Brain Initiative Cell Census Network and the Cell Atlas Network. And I could have titled this talk from BICCN to BICAN, actually. Those acronyms mean the Brain Initiative Cell Census Network. This was a phase of this project which was largely focused on mouse with pilot work in human and non-human primate. Moving to now the new phase, which is called the Cell Atlas Network, or BICAN, which is largely focused on human and non-human primate. So just to sort of quickly frame where we've been coming from, for years now within this cell census project, we've been trying to create a comprehensive map of the cellular organization of the brain. The very first portion of this was focused on one part of the brain, sort of the ENCODE model, pick one region of the brain and try to go very, very deep. This brought many different modalities together to try to understand how we could characterize cells. Many of you in the audience were part of this. Aviv, for example, was part of this. This culminated in a nice nature package to sort of set the paradigm for this. So fast forward 2 years. One more point I wanted to make on this, and this is really to feed off of what Aviv was saying at the beginning. You know, a cell atlas needs to incorporate all these different characteristics of the cells and the tissue organization. And so the output of looking at one region here was a hierarchically organized, transcriptionally based classification that captures all the different types of cells, that includes the types of cells, their gene expression profiles, their epigenomic signatures, their spatial organization, and we're starting to understand their cellular properties, their anatomy, their physiology, their connectivity, for example. So this is really what a cell atlas means in at least one part of the brain from the Brain Initiative's perspective. Now fast forward 2 years, and the scalability of some of these methods has allowed us to characterize the entire brain now in mouse. And a really good sort of heroic effort on the first attempt at doing a human brain. And this gives an answer to one of the questions that was already posed here of how many types of cells there are. With those similar types of criteria, there are about 5,000 discriminable types of cells in the mouse brain. The human brain, this was a sampling. Of course, it's 1,000 times bigger, so it's much harder to do. Looking at about 100 brain regions, we've gotten to about 3,000 types of cells that we've characterized so far. So really good progress, but not quite comprehensive at this point. In the mouse, this has also been a period where the spatial methods caught up. And so now we have not only a classification, we have a spatial map using MERFISH to describe the spatial organization of all of these types of cells as well. And that actually becomes an important criterion for whether this is really a cell type, its spatial organization. I wanted to give a little plug here. My colleagues at the Allen Institute provided me with this slide this morning, that tomorrow we're going to have a release of a first tool to be able to visualize this whole mouse brain data. This is called the Allen Brain Cell Atlas, or the ABC Atlas, that lets you navigate through the brain like the original Allen Brain Atlas, which was genes. Now you can look at cell types and can impute gene expression across cell types across the entire brain. So this is sort of where we're going with these things, a complete description of the cellular complexity across the brain. Okay, I want to move now to BICAN. So as I described, we have a very good draft atlas of the human, but it's by no means comprehensive. And so the next phase of the Brain Initiative, or BICAN, is aiming to try to really create a full description of the human brain. And the areas of focus for this particular project, a small number of projects were funded. These are very big projects. Each one of them is a consortium in its own right. are aiming to give a comprehensive coverage of the adult and human and non-human primate, and also development in mouse, non-human primate, and human. So these series of projects are leveraging now mostly multiomics, spatial transcriptomics, and a little bit of additional cellular characterization in some of the projects. The mouse actually allows you to go incredibly dense temporal coverage, which then you can map onto with sparser coverage in the other species. So unlike BICCN, there's a lot more centralization in this second level of the effort. There's an effort to centralize production of neurotypical brain specimens for use by the consortium. There are centralized sequencing centers, and there's centralized processing of the omics data in particular at the moment. So all of this data goes into the archives immediately for open access, and there are a series of not only the experimental groups, but also coordinating centers to be able to process the samples and the data through the whole ecosystem. We've really tried to set this up with the consortium focus on the outcomes and the products. So one outcome is published data. This is data that goes into the archives, increasingly annotated data. A second output is our standards, specifications, and SOPs. We want to make foundational references. We want to make, a la the human genome, the canonical cellular description for the field. And also software tools to be able to effectively visualize, mine that data, and of course scientific discovery with publications. A big focus of this is on anatomy. As I think Malte mentioned, location is extremely important in the brain, and it's very difficult to do in the human brain very accurately. So we've been trying to set up common coordinate frameworks. This is well known in the mouse. It's also well known in human neuroimaging. We need these references to be able to map samples into, and to be very, very precise about this, because a lot of variation happens as a function of position. We've been able to establish a common set of CCFs and also structural ontologies that all of the groups within the consortium will use. Hopefully will get adopted across the broader community soon as well. In order to really feed this, we had to go back to how to generate a very high quality set of human brain specimens that can feed this, and using the best optimized methods that are in the field at the moment. And so this has been actually quite a long effort to try to establish the best practices in this field and to get a series of brain banks to coordinate to use these in a similar fashion. Some of these things are fairly straightforward, like the proper freezing methods to be working for tissue histological preservation, slab photo documentation, things like that. Thinner slabbing, it's very difficult to do, but you can slab brains at less than 5 millimeters thickness, so that you have much better precision of what's in that actual specimen. Same thing for demographics, for general subject information, things like that, so we can capture all this metadata associated with these. One of the things that we've tried to do, if some of you may have experience with requesting tissue from brain bank and you get to obtain a piece of DLPFC, let's say, and you don't really know what it was. And so we tried to change this whole practice that we'll have a portal where there are slab images from all of these brains. The user can come in, they can document exactly what they took, they can associate a name with the thing, and this can actually be a tissue request from the tissue resources as well. So this is really grunt work to kind of set this thing up, but it's really important to be able to have high quality, consistent samples to work with across the whole consortium. So if I focus a little bit on the project that we're running, this is called HMBA, or the Human and Mammalian Brain Atlas. This is the big adult brain atlas project. We're trying to achieve comprehensive coverage across the entire human, macaque, and marmoset brain, using a combination of multiomics, spatial transcriptomics, and PAT-seq to do anatomical and physiological characterization of cells where we're able to. This really lets us take advantage of the benefits of the different species. So for example, we can do functional characterization of non-human primate brains and then analyze in that particular brain to try to understand the cellular basis of functional parcellation in the brain. We're also trying to cover individual variation. There are other projects in the consortium to do this even more broadly, to understand how robust this is, how consistent it is, how to explain variance across individuals, and the same across species as well. So this is a pretty big effort to try to pull all these things together. The three big outputs that we anticipate coming from this are whole brain cell type classifications, both within species and mapped across species to have a consensus classification across mammals. Spatial maps of the tissue architecture. This is an interesting challenge in scale-up. It's one thing to be able to scale up sequencing. The spatial methods to date don't scale quite as easily. And so this becomes much, much sparser sampling, but trying to get a map that covers most of the brain. And then finally, to put all these things together to have this integrated map of both the cellular, structural, and functional reference frameworks. I want to spend just a minute on that last point because this is really something that hasn't been done very much before, which is trying to bridge scales from the functional imaging scale to the cellular molecular scale. And we would really like to be able to relate functional architecture to structural architecture. First of all, there is a whole field of trying to parcellate, in particular, the neocortex on the basis of functional response properties. We'd like to be able to match these things so this becomes relevant for the imaging field. But also, these things are variable from individual to individual. So we all have a region that responds to faces. It's not in the same place in each individual. So if you really want to establish that relationship, you actually have to do it within the same specimen. So we can do that in the monkey. We're even trying to set up a hospice care donor program to do it in human, actually. It also requires us changing the way we prepare the tissue. So we need to be able to map a post-mortem brain into the functional imaging space. And to do that, we need to be able to capture a three-dimensional representation of the brain and all the slab images. You can reconstruct that volume and map that into the MRI space. This can be done either with a surface scan or with a short MR scan. And this is now really feasible to be able to take this brain, reconstruct that volume, map it, and then transfer the parcellations from an imaging parcellation back onto the slabs. So just to give you a little idea, this is the work of Eugenio Iglesias. He's taken this set of slab images from a brain, mapped them into the MR space, and projected back down the parcellations. So it's very difficult to do this robustly just from frozen slab images. And so this is really key to be able to map regions accurately. Okay, let me shift in the last little bit to discuss the ecosystem. So a lot of data and tools are available from all this work that we want to make available to the community at large. A paper that's a good guide to this just came out recently about the BICCN data ecosystem. So I would encourage you to look at this as sort of a source for understanding what all is available and how to access it. As we're moving forward, we're now really starting to think more about these reference classifications and a knowledge base to aggregate information around. And so this knowledge base really would be the source for our foundational references, our CCFs, our ontologies, our cellular and spatial maps that we want to be meaningful for the entire community. And by that I mean the functional imaging community, the cellular and circuit neuroscience community, the people trying to develop genetic tools or to understand connectivity, and of course external consortia, both disease consortia and consortia like the Human Cell Atlas, where we can really bring this as the brain reference into the Human Cell Atlas. And just want to end by saying we're kind of following the discrete model, if you will, of sort of treating these as types at the moment, but of course this can be modified over time, and following genomics as the model in many ways. We want to have a reference classification that has formal releases and a mechanism for versioning this thing, formal nomenclatures, tools to visualize the reference, tools to map against the reference, and this knowledge base to aggregate information about these cell types. So you get this increasingly rich ecosystem, very much like the genome, where you know so much about genes now. We should be able to do the same thing for cells. And then just one last little point, just to dovetail off of Phil de Jager's talk this morning. This thing is now ready to use as a baseline for disease. And I hope that many groups will start using this, maybe some of you in the audience. We've been using this for a project of our own called the Seattle Alzheimer's Disease Brain Cell Atlas, or CAD, to try to map cells along the progression of Alzheimer's pathology and understand if there are cellular vulnerabilities or cells that are particularly affected. We're able to map at this extremely high level of resolution. And then we can see that very selective types of cells are lost or vulnerable as a function of disease pathology, or that have disease states which are only seen in disease. And we can not only map that, we can see their trajectories and try to understand the earliest events in disease. So I think this will be an obvious use case for many in the field for the Human Cell Atlas as a whole. So with that, I just want to thank many, many people that have been involved in this. This is a little daunting to represent so many people's work. Thank you for all of the people in the BICCN, in BICIN, all my colleagues at the Allen Institute, the programmatic staff at NIH. That I want to in particular call out the program staff have been really helpful to orchestrate these whole things. NIA and all of our colleagues in our own new BICAM project. Thank you.
Okay. We have time for a couple of questions for Ed. He's going to start online.
Hi, we've got one from Kabita Baral in Calgary. She's wondering why macaque and marmoset, but not chimpanzee brains have been used.
So the choice of the non-human primate model organisms is largely because these are the dominant models in biomedical research. So that's a rationale for wanting to bring those into this fold. You can actually do some of these techniques in chimpanzee brain, but there's no active chimpanzee research that's going on now at this point. These are chimpanzees that are in colonies, end-of-life sort of colonies, that they get to live out their lives. If there's a serendipitous ability to harvest brains and put them in a bank, we can access those. But really that's only for comparative studies where you're trying to understand maybe what's specifically human. It's not really a broad resource that would serve a big community.
Yeah, hi. Hi. My name is Sid. I'm here in Toronto. I had a question about the postmortem brain. You said you could map it to MRI. Could you say more? Because one looks very functional, and you would need sort of data which is under some response where you're measuring MRI or fMRI, and postmortem seems to be not have that. So how is that bridge made, or what's the idea?
Yeah. So I have a two-part answer. So the first part is, in order to do this mapping at all, we need to be able to map a postmortem brain which has been removed from the skull into the same coordinate space. And this is very similar to taking a new MR and mapping it against a reference template. So that in itself is a challenge. The brain is deformed. If you have any photo documentation for most brain banks, it's not much. And so you need to have the surface imaging or a low-res MR and the slab images just to be able to do that. If you can do that, you can treat it like a new MR scan and map into that space. And then you have whatever parcellation is in that space in FreeSurf or a human connectome project, you can now map onto that particular brain specimen. Now, you don't have functional imaging in that particular brain specimen in that case. But it's much closer than just looking at slabs. The second part of the answer is we would actually like to be able to do that in individuals that have had function localizing fMRI. Now, in a macaque animal, you can do that as part of your experimental protocol. And so as part of our HMA-MBA project, we have brought in collaborators, Vindrik Frywald from Rockefeller, to do a whole series of scans that localize where different motor, perceptual, visual areas are. And now we can actually go into that very animal and try to see what's different about those areas versus surrounding areas. We'd like to do it in human too. And at the University of Washington, we're trying to set up a hospice care program where volunteers will undergo a similar function localizing scan, and then we'll look at their exact brain. Now, hopefully, if you could do that in a small number of individuals and establish that relationship, now you can predict it in a much larger group.
So one final question from Neil.
Great talk, Ed. Fascinating how you're approaching this. How much interest have you had from Neil Henderson, Edinburgh, from the clinical side in terms of using all this rich phenotypic cellular data to inform tracers, functional MRI readouts in humans? So I'm just interested how much leverage you've got there, because you have such a wealth of data, and functional MRI is changing all the time. So I just wondered that, how's it going that way in terms of translation?
Yeah, so I'd have to say it hasn't gone very far to date. Mostly because most of this single cell work is just dissected brain region. You say I did the middle temporal gyrus or I did V1. So you need to map it into that space. We did this over a decade ago with microarray data and created a big microarray map. And that did get quite a few functional imaging groups to try to take that and relate their functional imaging data to it. But that was a long time ago. That's a now somewhat antiquated technology. So I think that what we hope is that this approach will allow that to happen, and that many new people will think of different ways to use it, as you're suggesting there. But it kind of hasn't been possible yet.
So let's thank Ed again for a fantastic talk. Thanks.
Thank you.
Okay, and we're now going to move on to the first of two discussions that are going to form the meat, I suppose, of this session. And one of the things that I think is very clearly a theme throughout the talks we've heard this morning, indeed the whole Human Cell Atlas effort, is that there's a social component to this that is at least as important as the scientific component. There's a huge number of us in this room. We work in different places across the world. That has its own challenges. And in the next little part of the presentation, Chloe and Shyam, on screen, you're enormous in the room, Shyam, just so you're aware. You're absolutely dwarfing Chloe and I down here. Are going to talk about some of the work that they've done on really using the immune atlas, which is where Chloe's expertise is, as a test case for cross-tissue atlasing and the incorporation of diverse populations. So really looking forward to this conversation. So Chloe and I are going to sit here, and you're just going to be above us, Shyam. So we're going to try and look at you in as reasonable a way as possible with this sort of the Zoom offering. It is quite intimidating, I have to say.
Yeah. Shyam, people are taking picture. We're going to send you a picture of
Absolutely. Oh,
you above us.
You're becoming
I feel
smaller.
Like
no, don't.
I feel like you're looking above us.
So maybe just
away from the screen, so.
So maybe to begin with, Chloe and Shyam, do you just want to quickly introduce yourselves, and then we'll get on to the questions?
Absolutely. So I'm Chloe Villani. I'm one of the coordinator of the Immune Bio Network. I'm also part of the organizing committee. And I'm also helping spearhead the cell annotation platform, and I'm a big immune cell fan. And this is how I got to meet my friend Shyam.
Shyam, do you want to introduce yourself next?
So great to be here. And yeah, as Chloe mentioned, we've been working very closely together. I'm here, I guess, mainly in my capacity as co-lead for the Genetic Diversity Network. And as we'll talk about later, that's closely tied into the Immune Cell Atlas.
So maybe to begin with, why did the two of you decide to work together? Obviously, you get on well, but is there a particular reason why the Immune Atlas should be the spearhead for this incorporation of genetic diversity?
I mean, I'll point out that Shyam and I didn't know each other before the Human Cell Atlas endeavor, and so that's one of the really fun part of this community, get to meet people from around the world. And perhaps, Gary gets to lead the best atlas, but I think the immunologist fans are the friendliest and a really nice community. I'm just kidding. We're not competing here. In brief, I would say we both care tremendously about immune cells, but perhaps to kind of bring context, I could explain how we landed on blood, which may be obvious to some of you, but not perhaps to all. And so over the course of 1 year, we had several roadmap sessions, Zoom at different times of the day to accommodate the whole globe. And immune cells, as Aviv pointed out, are across the entire human body. And it was very tempting to just take all data sets out there and integrate them all at once, but realized this would not be feasible because at the end of the day, we all agreed as a community that we had a central challenge of annotation. We didn't even understood if we were annotating the cells the same way. And so what we ended up doing is splitting all immune cell data sets into 3 tiers of priorities for our community. First one being blood and lymphoid organ systems, second being mucosal burying organs, and then the rest. And then when we compiled data sets, it became very clear that we should just go with the blood because while it may be obvious to immunologists, it turned out that we realized we weren't even sure if we were approaching it the same way. And to start having that conversation, there was a need to define some common ground. We needed to have this conversation about annotation using the same data set. And it needed to be complex. It couldn't be a single data set. And it couldn't be just a million cells, because that's too easy. We wanted something that was very complex, very broad, to tackle the question of cell type definition, cell states, perturbation. And that's how we landed on blood. And through these meetings, I got to meet Shyam. And he told me more about the scope of the genetic diversity bio network and realized that they also had a lot of blood data. And ultimately, we need to make sure we align in our approach for annotation so that eventually we can really bring it all together. But I'll let Shyam expand on the scope of the genetic diversity bio network.
Thanks, Chloe. Yeah, maybe I'll zoom out a bit and then come back to your question, John. So the mission of the HCA GDN is to understand how all forms of human diversity manifest themselves. And when I say manifest themselves, in our case, it means manifest themselves as cell types, cell states, molecular properties of their cells, and so on. And the key axes of human diversity we're looking at are genetic diversity, that individual genetic variants like what Tuuli was talking about, genetic ancestry, sex, age, geography, environment, lifestyle, and so on, right? And of course, part and parcel of that, so there's a clear scientific and medical need for all of humanity, but also part and parcel of that is implementing or advancing the equity goals of the HCA, which means everyone should be included, represented, participating, and benefiting from the Atlas research, right? So that's broadly what the genetic diversity network wants to do. And I'll rephrase that now in mathematical terms, okay? And then that'll make it clear why connecting with the immune cell atlas is so logical, not just because, Chloe and I meet a lot at meetings and we get along well, but also is kind of inevitable, okay? So what the GDN is doing, you can say what the organ atlases are doing is they're calculating the mean at some level, the average property of each cell type, mostly, right? And what the genetic diversity network is doing is calculating the derivative around that mean, right? So which means how much variability is there about that mean. Now that means that anything we do in the GDN is predicated on what the organ atlases are doing, right? It's not logical for us to define the derivative independently of how people are defining the mean. And what that means in practice is that we have to use the cell type definitions of the organ atlases, cell state definitions, the metadata, actual metadata from the organ atlases, so that there's internal consistency and interpretability. So that's one thing, that's the second reason you can say. Third reason is that whenever you're calculating the derivative of something, you need more data than to just calculate the mean, right? And in our case, more data means we need a larger cohort size, as Tuuli was also alluding to. And where do you see the biggest cohort sizes right now? It's really in blood. Although it's amazing, Philip was showing this, I think 420 individuals' brain data sets, so that's massive as well. But still, blood is the largest right now. So for all these reasons, I think it's very, very logical for the GDN to come alongside and a little bit after the immune cell atlas.
So maybe just as a quick follow-up to that, how important is it the population that's used to define the mean? Because one of the criticisms in a lot of genomics research is that you start with a population derived from generally Western European ancestry, and then everything else kind of iterates off that. So do you think that here, are we making the same, or do we have the same challenges in our community about what that baseline is? And is that baseline itself diverse enough for what we want to do moving forward? I'm curious about your thoughts on this. Maybe Chloe to start, and then Shyam.
Well, given that's in the scope of Shyam, I'll let him answer first, yeah.
Okay, Shyam.
Thanks, Chloe. Great question, John. And yeah, I think if we go by the human genome sequencing paradigm, then the baseline or the mean is defined as some kind of Western reference, and then the variability is how different is everyone from that Western reference. And in a sense, it's a convenient or easy paradigm to implement, but may not be ideal. What we really aspire to in the Human Cell Atlas is something broader, where we incorporate diversity from the start in defining the mean. Or to put it differently, we say that there are different means in different parts of the world. the male and female means are different, European, African, Asian, etc., Australian means are different. And the young and old means are different, and so on. So in that sense, we're hoping that it will be a different paradigm here in the Human Cell Atlas.
But if you're asking us about this first iteration, we had to go with what we have, right? And as a minimal viable product, it won't be perfect. But actually, it's pretty amazing that we already have some diversity, because a lot of us contributed data set that were not published. Shyam did, I did, and other colleagues. And so while it's not truly diverse, it does have diverse population represented. And that will help us, start defining these gaps, although there's a lot of ongoing effort in Shyam's network. And so as part of our first common minimal viable product, we really wanted to work on just annotation, which seems obvious, but it's really not obvious. And figuring out how to engage our community in the process. Ideally, you would want the whole community to take part to every steps of the process, but it's actually not super feasible. So a smaller group of us are working together to create an integrated object, which we made sure that would be as diverse as possible. And then everyone in the community will get a stab at our annotation. Everyone has a say. There's going to be if you want to hear more, we're going to talk about it tomorrow during lunch in room CR2. It is not on the schedule. But, if you're not on the Immune Bio Network email list, I'm just telling you, 12:30 CR2. But there will be a broad announcement where you can take part of it wherever you are in the world, because it's going to be also virtual. And if together we can achieve some framework for defining annotation, which we'll be doing in collaboration with the Cell Annotation Platform, then Shyam and his bio network will be able to carry on with expanding the diversity.
Cool.
Yeah.
So what were some of the technical challenges then associated with that? Is it really just about wrangling the data, getting it into the right format? What were some of the logistical challenges that you faced in getting that baseline first set of annotations?
I'll take one, and then I'll let Shyam take one. I thought we were on top of it, because we had, as a bio network, spent time the year before figuring out which data set we would focus on with the PBMC. So we had them. But we didn't realize how much of a challenge it would be to align our metadata across data sets. Someone from Indiana, from breast, mentioned, they had particular data set that were of high quality. We had the equivalent, although blood is much easier, but even that. And the metadata definitions were not exactly the same across data sets. And so doing that alignment took a long time. I encourage you to go see the poster from the blood integration team. But for the next wave, our lesson learned are being propelled in a new tiering of metadata schema, where we're encouraging people to go into simple tier 1, which is more technical, and tier 2 biological, which we hope will help. And that information will be circulated to the community. So that was a surprising technical challenge. We thought it would kind of just get going quickly. Shyam, do you want to add to this?
Sure. I think the technical and process challenges or learnings go hand in hand, you could say. Just to pick up on what Chloe said about metadata, perhaps now that we know a little bit better what metadata we need, and the HCA is polling everyone saying, what are the metadata for your network? The next step would be to say that at the point of submission, we give the submitter a list of harmonized metadata fields that they need to fill in, so that then later on, that's much easier than later on chasing them, the postdoc may have left and so on. So I would say that's a process learning on that. In terms of a more technical learning, we were a bit surprised, but in hindsight, maybe we should have known it, that we focused a lot in our own network on harmonizing protocols. And by that, I mean the bench protocol across countries, across labs. And then we were quite happy about that. We did a good job of it. But then we still found substantial batch effects across labs, and different labs in different countries. And our understanding now, based on some internal controls, is that those batch effects come not from our bench protocol per se, a molecular protocol, but from how the samples were collected. And it seems across studies now, actually, that is perhaps the major source of technical variation, rather than what you do with the cells once you get them at the bench. So I think more attention needs to be focused on harmonizing that.
And so maybe just to close this out, what about the people aspect? The ability to manage such a complicated project across so many different time zones, logistical, technical challenges. What's worked well there, and what do you think we could learn about how to do this better as we move forward? Because this is only going to grow in scale.
Shyam, do you want to go first?
Sure. I think there's definitely that time zone challenge and getting people to show up for meetings challenge. What we've found is that, people will, if you try hard enough, people will show up for meetings, at least some subset of them. But to keep them engaged on a longer term, on a more continuous basis, right, rather than having them just show up and disappear and be disappointed and disappear, you do need concrete goals and you need to show progress. If you just have meetings and you're not somehow showing progress, then people will disappear if they feel that this is just talking and not going anywhere. That's, I guess, our main thing. And then the second thing is that it helps a lot to organize workshops. So that, no, it's not just that you make concrete technical progress in the workshops where people show up in person. It's also that, it's not easy to get people to answer emails or messages or really try to communicate or connect with others if they have only met them on Zoom. So I think that is a very key aspect of any successful collaboration, that in-person concentrated interactions at workshops.
Just to add a few things, we've been making a conscious effort to actually organize meetings, switching times, So I take meeting 10 p.m. to midnight, and Shyam does the same. And we kind of take turns so that everybody can really join. And we are open for feedback, how we can best engage the community. We've been trying to find ways so that everybody can contribute and pitch in. With the first idea being this annotation jamboree that would have virtual and an in-person component. But please find us. Shyam will be here during the meeting. He's arriving tonight, and I'm here as well throughout the meeting. So we're looking for feedback. But to Shyam's point, like these in-person meetings, they make a huge difference in forging these connections. Yeah.
Absolutely. So on that note, as we are all here together, so let's thank Shyam and Chloe. Lovely discussion. And then we'll move on to the second discussion topic, which is kind of flows very naturally here, which is how do we actually define Atlas 1.0? How do we get to the end? And now we're joined by more people on the screen. And Aviv up here. Okay. Hello, Dana. You're. These have wheels. You are all large on the screen. You are now only you're 1/4 of the size you were, Shyam, though. So relatively a lot smaller. Yeah. So less intimidating, though. Given the people on this panel, I'm not sure the ability to keep to time is going to be trivial, but we'll just stop at some point. Excellent. I'll take that up with waffling. Okay, so let's begin with what is a totally trivial question. How do we define version 1 of the Human Cell Atlas? Aviv.
I thought you didn't want me to speak at length. I think the way we set it up for ourselves was one modality at first, RNA profiles. Multiple data sets in each organ or system, separated still between the system, with the exceptions of things like the immune system, where you often come across, but not yet, the gut and the liver and the lungs and so on, all combined together. So those are maybe the three simplest definitions around it, but done in such a way that between those, there is a relation to the anatomical position, harmonized names for cell groups at the very least, as well as ideally, not in a minimal viable product, but in a full first version, as well as within that context, also having other ways of looking at the cells besides categorize them into harsh categories. And, you know, I'm always jealous of the brain people. I don't know where Ed is right now, but I'm very jealous of you because I think actually the strict categories and the hierarchy works extremely well in the brain, but once you move out of the brain, actually it stops working. You have a much more fluid situation for many things because actually neurons and other cells behave quite differently from each other. So that's a thing that sounds a little fuzzy because I don't think we understand it fully. I will say that I think for some systems and networks, the data are there to suggest that even in the first version, we can be a lot more ambitious. There's either spatial data that allows a tight connection, or there could be better anatomical sampling and a good understanding of a coordinate framework, so you can go down that road, or there is more multimodality in the data. And so what I said is kind of the minimal of a version 1, but in some cases you would go into deeper than that because the data are available and we should be moving along.
Okay, cool. So maybe turning to Sarah then, we've got this sort of outline of what should be in HCA 1.0, but how will we be able to find it? How will people actually be able to use it? Do we have sort of ideas of how we're going to make it accessible to the community?
Yeah, that's a great point, John. Can you hear me?
Yep.
Yeah, it's a great point and, you know, incredibly important, obviously, in order to get the benefits of HCA out to the broad community, the biomedical community, and also the global community, you know, including low and middle income countries, including people all over the world. And what we need for that is, having all the data deposited in rapidly and organized. And that's really what the data coordination platform was and is about. And, the mechanisms for managed access data are being developed now, which is amazing. And where there's more development and where there's ongoing activities, as you've heard and as you'll hear more on day 2 or 3, I think is on, or you'll hear over the next days, is the integration of data. And then also where as a community, you know, for the computational community where we need to put more effort is the portal development, and those efforts are ongoing, you know, to make the data kind of nicely accessible, visualized, queryable, and so on. And, that is absolutely key from the point of view of benefiting from all this data that's being generated, and, that Aviv talked about, and version 1.0, and then we'll go to version 2.0, 3.0, et cetera. And in parallel, we really need to have the accessibility of the data in a nice, not just the raw data, but the different levels, the integrated data, and then in a nice visualization, queryable, and so on, so that people can interact with the data and ask for their favorite cells, their favorite tissues, their favorite, ultimately tissue niches, and so on.
Chloe?
Gene.
Yeah, I just want to add a few more points about Sarah. So John Randell is going to show us some highlights of the portal next, which will be really key in enabling tailoring of the different data sets accessible for every bio network. So it's not only about the integrated object, but it's also about the individual data sets that were integrated, right? It's really important for data interpretation. We're also working in securing raw data because we need to future-proof our atlas. So all of these data sets that will be individually integrated, actually together integrated, the raw data sets will be available. And the portal will also link to different visualization tools, but as well as to our Image Atlas cell annotation portal. So to me, part of what defines a version 1 of the atlas is achieving a community-wide consensus annotation for your minimal viable product, right? That's, to me, what helps define, did we achieve a version 1, regardless of which data sets they're being integrated? And so the cell annotation platforms and Evan Bidders then is here. You can touch base with them throughout the meeting. The team will be working with every bio network to define cell cards, once you have achieved some forms of consensus annotation, and as Aviv mentioned in her introductory remarks, this is a living product, because we know these annotations will evolve over time, and so that too will be tied to the portal and will help us move forward and define the gaps, right, for the next iteration.
Aviv?
I do think it's really important. Something came up both in Malte's presentation and in Ed's. Sometimes people see the first version and that becomes kind of in their brain, that becomes the thing. This is really just a first version. And it can change both in the sense that Malte described, you just add more and as a result you iterate through it. And the goal is for these iterations to become fast and furious. And it can be also qualitatively different. That's something that Ed really illustrated nicely. For example, in the Brain Initiative, the first version for human is truly just cell profiles, RNA profiles. For mouse, it's very different, but for human. And now it's going to be this multi-layered thing. The same is going to apply here. And so it's really crucial for us not to become too attached to the first thing that we got, because that's not really the end point, it's just the starting point. And it is important to start and integrate for the reason that Chloe said, that we need to make sure the community, it's their thing. It's not any one group's or researchers' baby, it's everyone's baby. And so they have to all be owners of it. And for this, we really need to do the integration.
So one of the things that struck me from what we've heard now from Malte's presentation as well is that there are different techniques are needed for integrating different data from different cell types, different atlases. So I'm curious, maybe to Dana and then to Shyam, one of the key uses of this atlas is how we integrate it and how it's interrogated. So are we also going to need a variety of different tools for how individual groups are going to take their data and interrogate that in the light of the human cell atlas? So maybe Dana first.
So yeah, first I'd like to sort of follow up exactly on what Aviv said, that, right now, the thing that Malte described, it's really defined as a minimum viable product. And, the atlas is not one big giant UMAP of single cells projected on 2 dimensions. And the atlas is also not just, cell types, which, as Aviv said, are very, very limited. Now, this has been sort of driving our work, because even integrating these large data sets and having some harmonized cell types to them is incredibly hard. But then, and I know that we have a lot of guests here from the ML community, that's not the end goal. And I think we've really narrowed in on this goal too much. And, for ML folks, it's really great. It's a very simple benchmark. Is my integration capturing cell types? But we've seen that this integration of just cell type coarse labeling, or even fine cell type labeling, removes a lot of important variation. So I think what we need to define now as a community, if we want the cell type to work, if we want atlas integration to work, if we want the atlas to be usable by people for biological discovery, is to define how we want to use it, because integration, there's no free lunch, it has trade-offs, and a good integration is how you want to use it, what queries, and we need to move on to what I call sufficient statistics, because sparse millions and tens of millions of sparse single cells is not as informative. But the data is, we need to extract what is our queries, what is our sufficient statistics, gene programs. We forgot that genes matter. In fact, some of the embeddings lose information about genes. And so what we really need to focus now on a community is to define what are our queries and what are the sufficient statistics that we need to make these queries powerful, accurate, and efficient.
And do you think that those will be the same queries and the same statistical tools for every cell type, for every data type, or are we going to have a suite of querying tools depending upon what we're looking at?
We are going to have an evolving suite, but the nice thing is that they're not mutually exclusive. You can say, okay, here is, and they also take less space, and you can compute them once and then use them. So you can even have one atlas and 10, 20, as many types of sufficient statistics that you need, each used for their query. Different atlases will need different sufficient statistics based on the queries that they do. But I think that as a community, once you figure out how to do it, the information can translate perhaps with minor variations on the theme to the specifics of a specific tissue network. So I think there's going to be a lot of them. There's going to be some customization and a lot of sharing.
Shyam?
So we need to get started on this, and we haven't really.
Now Shyam.
Yeah. John, you brought up some very good questions. So I want to pick up on that theme, which is the answer can be different for different biological networks. It won't be one size fits all, right? And what does that translate to in terms of implementation? That at least from my perspective, maybe biased being a bio-network coordinator, is that the bio-network coordinators need to be in at the start of the whole journey and the methodology development and study design and so on for data integration, because they know what works, what does not work, right? And what worked for one bio-network may not work for another bio-network. Now that's a very vague statement. Let me make it more precise, right? So some simple things we think we know, right? That if you do data integration, you integrate 3 data sets, then your clustering in the integrated object is better because you have more cells, you more precisely define cell states in that integrated object. And in fact, we take that as the truth, that the integrated object is true, and what the individual studies did with their individual data sets is less accurate because a smaller data set, right? But any logic in science, when you extrapolate it to a data set that's way bigger than what you have experience with, can fall apart. So if you integrate 50 data sets with profound batch effects, profound technical variation, you'll end up, and I know UMAPs are much maligned, but let's go with it for a minute. You'll end up with a UMAP that looks like a blob. It has no features, and it has no features for a reason that you have actually blurred out the biological variations between cell subtypes by trying to overlap things that are very, very different technically, right? So that simple conventional wisdom of data integration is better than analyzing individual data sets can fall apart if you integrate the data the wrong way or integrate the data the way you were able to do it for small data sets, right? So I think it's very much a journey, and for every biological network, this number of different cohorts, number of different data sets is different. The amount of batch variation across data sets is different. The scientific question you're asking in that biological networks is also different because you may be looking at broad differences between, let's say CDC1s and, monocytes, or you may be looking at very subtle differences between male and female, for example, right? And then your data integration method that works for question A may not work for question B. So I would say there's a lot of learning to do when we scale up to this size.
So building on that, maybe, Aviv, you mentioned at the beginning foundational models, and this is obviously extremely important in a huge variety of contexts in LLMs and other areas as well. So maybe for those who are from the machine learning community who are here and maybe being exposed to HCA for the first time, what do you think of as a foundational model in the context of the Human Cell Atlas? Is it just the data? Is there a model underpinning it? What is a foundational model in an HCA context? Maybe start with Aviv.
Yeah, I'll answer, and I'll also say that in the context of the question you asked Dana and Shyam. I think always when you think about a model or a representation in this particular case, you want to think about the representation and you want to think about what you want to do with it later. And those two things, at least in my mind, are inextricably connected in interesting ways with each other. What I think is interesting about foundational models is the sense that if you've seen enough, if you let the initial model in pre-training and training be exposed to a large enough part of your universe, and no one ever knows what that large enough part is, it will encode something about this universe that would make it very empowered to do more things for you than you would have explicitly expected it to do ahead of time. So for example, it could end up having predictive power. One of the things we've always hoped for in HCA is, what if a model could suggest for us a cellular profile that we've never observed? And would say something like that should exist because the scope of this universe that I've now encoded is more than just a recording of the data that I've showed you. That is what is interesting about these kinds of foundational models and why they are foundational rather than just a descriptive model of the information that I provided. That in the process of encoding that world and representing it, it spans a space that includes possibilities that were not shown actually in the training, and then it would generalize to new questions. So that's the general premise why I think it is particularly exciting for the Human Cell Atlas, and also why we probably are at a point that across at least the systems that have been looked at quite deeply, sufficient data have been collected that should make this not a fool's errand. There's a there that would take you kind of beyond the place of the data itself. Then beyond that, you do have to think about what are interesting questions to ask. And in that, I think what behooves all of us is to first of all take the questions that we kind of know are interesting for us, but we don't have the answers for them. But you want to think beyond that. You want to think about questions you haven't asked yet. That is what was fun with single-cell genomics in the beginning, and now with spatial methods, I think should be fun again. That it's not just asking the old questions and we just have new methods, new tools, new techniques, new data, but because of the existence of this new data and these new tools, we can actually ask questions we haven't asked before. Not just get better answers, but ask different questions. That happened with single-cell genomics. There's questions we ask there that people didn't ask before in biology. There should be more generations of questions like that, and that's where this going back and forth between a foundational model and the capacities of what you can eke out of it becomes particularly exciting biologically.
Any other thoughts? Anyone else want to jump in? Dana?
So, I think actually the issue of foundational models is exactly why having a consortium like the HCA is so great, because, I had the fortunate luck of having Fabian visit my group personally last week, and he showed some beautiful work that you get nothing until you have tens of millions of cells. It's all gobbledygook. But again, the questions, the queries are critical, because we know that foundational models can hallucinate, and I'm waiting to see the hallucinations and the total absolute BS that these things will spit out if we don't train them properly. Foundational models is all about training it properly. Therefore, that needs to be done hand in hand, biologist and machine learning expert, so that actually the latent will represent real biology, so that these new states will be meaningful. I mean, even with foundational models, the moment that we gave them math, we gave them logic, they started to be much better. So I think we really have to think, what are the questions? What do we want to learn about biology? And how can we train these models so that we will actually get that and not bullshit hallucinations?
But is that not more about fine-tuning rather than the foundational model? There's an argument that once you have enough, you just need a sufficient data.
No. Foundational models isn't just, throwing a crap ton of data. It is how you define the training. It is how you define your target function. You can have a crap load of data, but if you don't train for the right target, you'll still have BS, at least as far as what I know about foundational models. And I'm not a big expert here, but this is sort of my take. It's all about how you train them and not just how much data you spill in.
But John is right that in the fine-tuning, you can kind of target your model in a direction that is particularly relevant to your question. So maybe both of you are right. But I do want to make a comment on hallucination.
Okay.
So I think hallucination is particularly interesting about these models for two reasons. First of all, that's where creativity starts existing, right? When things get hallucinated. And that's interesting for us as scientists. And second, there's two levels at which you can think of cells. There's the capacity of cells to be as they are in nature, and there is the capacity to drive them to be things that might be not entirely natural, but may be quite useful. And so that capacity to hallucinate could be quite interesting. And if the model is equipped, not just with representation of data that it saw, but for example, as you said, with mechanisms, for example, which is something that I think we are going to grow into, then there is the possibility that this becomes actually engineerable, which would be kind of fun.
Chloe and then Shyam.
We can add a biological spin to the hallucination, and it opens up a new path that biologists perhaps have not even suspected. And so if there's enough confidence in some of these predictions, as I can put my immunologist hat, it gets me to think twice about a concept that I thought was already defined. And so that's actually really important, and it's one of the power of this consortium, right, to kind of open new ways.
Shyam?
I want to add a couple of things to the mix, going off what Dana said, which is that we get excited about foundation models, deep learning, and so on, because it's kind of new and sexy. What we don't get excited about is simply cleaning up the data, which takes a lot of time, exploratory data analysis and QA.
Boring.
Exactly. Well, but it has a much bigger
colleagues
effect on the outcome, right? So reviewers don't like to see it, journal editors don't like to see it, but in our experience, that actually matters more than which deep learning algorithm you use. But of course, fine-tuning your model, your deep learning model, does definitely help, and everyone sees that. I want to add one more point about this, not just data cleaning, but feature extraction, feature selection, and so on matters. So we assume that your deep learning method is going to, by some magic, do the correct feature extraction and identify the correct features, if we could only give that model enough training data, right? But in our experience on something, I would say much simpler, something as simple as simply aligning the shapes, clustering the shapes of cell nuclei, simple two-dimensional contours, right? How easy can it get? Deep learning methods don't do as good a job of aligning or,
They can see your text, just so you know.
So people are aware, we can see your messages that you're sending to one another, so that you have to use Dana's trying to use both a physical and a virtual hand, which I am trying to ignore to keep to time, but I don't have a mute button though, so I suspect no matter what I say, you're going to say something. So go, Dana.
All right, over to you.
Just to say that hallucinations aren't a good creative things in which we learn new valuable stuff. Hallucinations, in my definition, is when the latent is total BS. I mean, these are matrices, and it only works if it's capturing a real abstract notion. Now, I do hope foundational models will really give us big aha moments if we do them correctly. And following on Shyam, I have a saying: garbage in, garbage out, and he's on task for sure. But we have to distinguish between any bullshit a foundational model spills out and said, oh, that's interesting, and take everything with a grain of salt, and new things and new abstractions is welcome. BS is not, and there's danger of BS.
So, okay, so we're going to try and filter out your use a profanity filter moving forward, I think. So Fabian is desperately trying to say something. So
you couldn't go like this for long.
Let's let him have one question in the audience from Fabian, just a comment.
I don't even want to keep the discussion going without profanity, anything. We have a session coming up, breakout session, quick block on profanity, but foundation models, and basically all of these discussions. I took a bunch of notes and
Dana is invited to discuss hallucinations there.
Yeah, let's maybe even the real Dana if the flight goes. So this is just the thing. Hope to see you there.
Excellent. Thank you for the plug. So let's finish with just from each of you turning away from hallucinations to maybe the area of health and disease where you think HCA is going to have the biggest impact, and it can be outside your area, but where this compendia of cell types, locations, where is it really going to have an impact on the lives of people across the world? So maybe Sarah, starting with you on that.
Thanks, thanks, John. I mean, we've already had a massive impact with COVID, right? In terms of just using healthy HCA for mapping viral entry receptors around the body, and that's COVID, but it can equally, be for avian influenza or whatever the next threat is, and then inform basically on that. I want to also plug Drug to Cell, which will go online, which is online on bioRxiv and will come out in Nature on the 16th of July with our Heart Cell Atlas collaboration, and a couple of the authors are there, James Cranley and Kazumasa Kanemaru from my lab, and that's basically doing the same thing as for the viral entry receptors, just for drug targets. And Aviv, is sort of always been leading the way with the genetic mapping of cells. So even just having the healthy human cell atlas, the healthy reference cells, plain and simple, has already had a massive impact. But obviously, there's also the, acting as a reference for what changes in disease, what changes in treatment, what changes in any sort of longitudinal perturbation. And human cells is having and will have, a further impact in that sense. John, you can talk about Milo-2 and human reference atlas calculations. So I'll hand over to you for that and stop there.
Okay, well, I'm not going to speak in this session or provide any insight. I'm just going to hand it over to someone else to provide meaningful insight. Shyam, you next. Where in a disease context is HCA going to have a real impact?
Certainly, I'll say from the GDN perspective, HCA as a whole, diagnostics is very important. Very few people are talking about single-cell diagnostics right now, meaning using single-cell assays themselves as a diagnostic. But certainly these single-cell assays can give you a healthy baseline based on which you can do non-single-cell or more traditional diagnostics more accurately, right? So I think single-cell data are going to tell us more and more, like a better thermometer, a better readout, or tricorder or something, right? So I think diagnostics will benefit hugely from the markers and correlations we identify. The other thing is drug development. So as Tuuli pointed out, QTLs are really important for going from variant to function and taking our GWAS hits and telling us which genes are relevant and why. So single cell QTLs will advance the field substantially, I think, and then help us interpret GWAS better and design better drugs.
Dana, where's it going to have an impact?
So here I'm actually optimistic, contrary to myself. I think it's already having, and it's going to have an impact everywhere on every disease. In regenerative medicine, it's having huge impact on cancer, and this impact will only grow. I think this is going to be transformative, and here I'm actually incredibly optimistic.
Awesome. I love that. Chloe.
Perhaps to frame the answer to my question, I think it's helpful to reflect on why HCA community is so special, right? Why is it going to be so impactful on the biomedical community and just the broader community in general? And that's because of how a consortium is built. We're not driven by a single funder or a single disease mission. It doesn't matter who funds us, as Aviv mentioned, right? It's more of a community of people who care about this mission. We're one of the most diverse community. We have graphical representation across over 95 countries. We're covering the whole body. And this amazing community that represents every single field, from the actual biology to methods development, is really what's going to empower us to have an impact on everything that was mentioned, whether it's diagnostic, whether it's, and as Dana mentioned, it's already having some impact on finding drivers of disease, finding potential therapeutic targets. I myself focus on trying to figure out toxicities, side effects to treatment so we can improve the life of cancer patient. But globally, I think there's no disease out of reach. It's also important in terms of just revisiting our understanding of what's healthy and normal. And so this community per se is extremely special, and it can really achieve anything at once. And we don't need to have the final atlas to already have an impact. Even these minimal viable products are already helpful to spearhead some new findings. Yeah.
So appropriately, Sarah, and then we'll close with Aviv. Sarah?
Yeah, and so one thing that, so there've been a lot of great comments. One thing that I don't think has been mentioned is the impact for regenerative medicine, kind of potentially in the long term, is both from the point of view of having the kind of reference healthy cells, the developmental pathways, and then also, as we've discussed kind of indirectly with the foundation models and the hallucinations, the gene regulatory networks and kind of regulatory relationships, is all of those pieces of information are invaluable for benchmarking and designing cells in vitro. And so that's potentially also a long-term health benefit, both in terms of sort of making cells to test in the dish for drugs, toxicity, et cetera, but potentially in the long term as therapeutics themselves.
Awesome. And Aviv to close.
To keep it short, everything, everywhere, all at once.
Okay, short and sweet and not at all ambitious. Excellent. Good. So let's thank the panel members again. and we're going to close the session with an update from John on how we can actually begin to think of interrogating and looking at these data. So I'm really excited by this, John.
Yes, thank you. Hello, everyone. I'm John Randell from the HCA Executive Office. I was actually asked to give you a preview of two exciting events coming up, one of which is lunch. So lunch will be available in a few minutes. You'll pick it up outside. It's very portable. As Gary mentioned at the beginning of the day, you can take your lunch anywhere in the building. I would just point out that if you go up to the top of the elevator, there's actually a room with some cafe tables where you can sit at. There's also a really nice patio. I'm told it's a beautiful day out. So I encourage you to use the patio if you wish and sit in the chairs out there as well. So that's lunch. If you're attending the ethics working group meeting, that is in room CR2. Your lunch is available outside the room, so you'll just go directly to the room. A few of my colleagues will be up there to point you to CR2. You can just get your lunch there and then start that meeting as well, and I'll be joining you shortly. So the other major development that I'm really excited to tell you about today is the forthcoming release of what we're calling the HCA Data Portal 2.0. I was really glad that data access came out in the last session, because that's something we had a lot of conversation about data generation and integration and analysis and so forth, but data access is equally important, as we heard, for transparency, for reproducibility, and particularly for sort of equitable participation and benefit from HCA. And so, as HCA evolves, the data portals, both the main HCA data portal and all of the associated community portals, will need to evolve with it. And so that's it's a very exciting chance to tell you about what's coming up by the end of this summer, but then again, continuing to evolve into the future. This is not my work. I just want to say that right up front. I've sort of been part of the large group of people providing input and feedback onto the evolving portal. It's primarily the work of Clever Canary, which is an organization that does product development, UI design, and so forth, particularly around genomics. Particularly would point out Dave Rogers taking the lead on this redesign of the data portal. He's been supported by the HCA data wranglers at EBI and UCSC. Again, getting a lot of feedback from the organizing committee, from the biological network coordinators, from the executive office, and I'm sure many others that I'm not even aware of, and of course pointing out that both the development of Data Portal 1 and the new Data Portal 2.0 are supported by CZI. So the data portal lives at data.humancellatlas.org. Hopefully this is familiar to most of you. You can see here some of the stats in terms of cells and data that are currently already available through the data portal. But the idea of this redesign is really to organize the HCA data portal around the concept of these tissue atlases that we've been hearing about today. And so I'm going to show you some mock-ups of what this is evolving to look like. I just want to be clear that this is not yet live. It's still under development, and in particular, the terminology, the content, all of that is subject to change. But certainly, we definitely want feedback, and I think in particular want to know what you would like the data portal to do for you. And so certainly talk to the members of the staff, talk to members of the organizing committee, the biological network coordinators, if you have ideas, and we'll make sure that those get passed on. So this is a mock-up of the new Data Portal 2.0 homepage. And what you'll see here is that, as before, you can find easy access to the data browser. There'll be a more visible button for contributing data, and we're also working on some protocols and approaches to streamlining data contribution. But what's new is really the organization around the atlases. You see those down here, and I know that there are 15 here. We're not canceling 3 of you. Again, this is very much a work in progress. So I'll actually begin with the Data Explorer. So if you just simply want to go to the data browser in the data portal, then that has also been redesigned to sort of increase navigability and so forth. You see the filters here on the left and so forth. I particularly want to point out that we're experimenting with ways to tag these data sets according to what atlases they have been used for. So you see, for example, here some, again, this is just mock-ups, but you can see here that the indications of where the biological networks that they're associated with, as well as the atlases that they are part of. Again, this is all mock-up data. So that's the data browser itself. However, the other way of exploring the data then, it goes back to the atlas-based view. And I'm going to use the lung atlas as an example here. As we heard, it's the first sort of published atlas that came out last month. So if you were to click on lung down here, it would take you to a homepage for the lung biological network. And you see a number of categories of information here. You see a general description of the network, some stats over here, contact information for the network coordinators, And then the atlases that have been produced by that network to date. I'll get back to this in a second, but I also want to point out up here there's a tab for all HCA lung data sets, whether or not they've actually been integrated into an atlas yet or not. What that page would look like is, you can kind of imagine, something like this, again, just showing all of the HCA lung data sets in the collection. Going back to the lung network homepage, though, you see down here that the list of atlas here, there could be different atlases that are portrayed here. It could be different successive versions of a particular atlas. You could have disease atlases that are associated with this biological network. There could be sub-atlases. So for example, in the case of immune, we're starting with blood. And so that would be sort of a sub-atlas, if I can call it that. And so if you click on this, then you arrive at an atlas homepage. And here's where you can see what we're currently calling component atlases, probably not the best name. Again, I'm still working with terminology. But for those of you who have read this paper, and the publication is linked up here, you know that there is both sort of a full atlas, which includes sort of disease cohorts, but also the core healthy atlas down here. So you would actually be able to view both of these here. As the sort of milieu of portals evolves, and as the atlas becomes more and more available throughout different visualization portals, then that information would also be linked to. So for right now, we have links to the cell by gene collections for LCA, but then as more portals make this available, we would also then add those as well. And again, contact information on the right. And then again, you can click on a particular sort of quote-unquote component atlas and get the list of data sets for that atlas, which I think just right off the bat gives you a lot more transparency in terms of what data is being used to integrate which atlas. So I know that was very quick. I didn't have a lot of time, but that's just to give you a flavor of how we are redesigning the HCA data portal around this atlas concept. Again, really want to hear your ideas for both the atlas itself as well as sort of features that you'd like to see incorporated. So I will end there, and I think we just want to thank all the speakers for the morning. John, anything else?
Unless there are any questions for John R. Any questions about the portal? Just want to have if people have questions about that. Yes.
Hi, we talked a lot about donor diversity, but there's no basically visual of like donor metadata apart from like disease healthy, or how many individuals does every study includes. I see that there is a sample level which is similar to what the data portal is right now, but I wonder if like there's more efforts towards also collecting a variety of like donor information that is simply like not healthy and disease. Yeah.
That's a great question. I think Chloe mentioned in passing that we're also looking at the metadata schema for HCA and thinking about, she mentioned a tier 1 and a tier 2. Tier 1 is largely technical and tier 2 is sort of biological, but that also includes a lot of information about, biological information about the donor, including all the different axes of diversity that Shyam also highlighted as well. That's still under development. That's one of the things that we'll be looking at to develop further in this fall, but would also be, to the extent that it's open access, would be available through the data portal, and then as managed access also rolls out, a lot of those data may be managed access, and they would also be available here too.
And then question down bottom left from Malta.
Thanks. I think the overview looks really cool, especially like the focus on kind of openness of this is really important. I wonder though, because you're focusing now on putting kind of references or integrations on there, it seems to me what's missing is a link to the reproducibility of the code. To kind of not only have the data freely available, but also something that people understand what happened here, and something that, so I feel like that may need to be included.
There was, I know I went through this very quickly, there was the beginning of a field for where the code is available. And so as thinking about how to make that as transparent as possible, we certainly want to incorporate that into the portal. It's there, I just went past it very quickly. Yeah, thanks.
Okay. If there are no more questions, then let's thank John and all the speakers in the session again, and it's lunch.